Check. Hello, hello, check. 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 Hello. Check. Hello, hello, check.
Check.
Namaskar, Jai Jagannath. A very good morning, ladies and gentlemen. A very warm welcome to the third day of AgriVision 2025. ସମସ୍ତଙ୍କୁ ପୁଣି ଥରେ ନମସ୍କାର ଆପଣ ସମସ୍ତଙ୍କୁ ଶ୍ରଦ୍ଧା ଏବଂ ସମ୍ମାନର ସହିତ ସ୍ୱାଗତ ଜଣାଇବାକୁ ଚାହିଁବି ଆଜିର ତୃତୀୟ ଦିନ ଶେଷ ଦିନ AgriVision 2025ର ମୁଁ ଚାହିଁବି ଗୋଟେ ଜୋରଦାର ତାଳି ସହିତ ଆରମ୍ଭ କରିବା ଯେହେତୁ ଆମ ସହିତ ଆମ ଗହଣରେ କିଛି ଚାଷୀ ବନ୍ଧୁମାନେ ଉପସ୍ଥିତ ଅଛନ୍ତି ଭାଇ ଭଉଣୀମାନେ ଉପସ୍ଥିତ ଅଛନ୍ତି ଅନେକ ଅନେକ ସ୍ୱାଗତ ଆପଣଙ୍କୁ ଆଜି ପ୍ଲେନେରୀ ସେସନ୍ସ ଯେଉଁ ବିଶେଷଜ୍ଞମାନେ ଆମ ଗହଣରେ ରହିଛନ୍ତି ସେମାନେ ପ୍ରେଜେଣ୍ଟ କରିବେ ଆପଣମାନଙ୍କ ସମ୍ମୁଖରେ ସେମାନଙ୍କର ରିସର୍ଚ୍ଚ ଏରିଆକୁ ନେଇ ଯେଉଁ ଏରିଆରେ ସେମାନେ କାମ କରୁଛନ୍ତି ଯେଉଁ କ୍ଷେତ୍ରରେ ସେମାନେ କାମ କରୁଛନ୍ତି ବର୍ତ୍ତମାନ ପ୍ରଥମ ସ୍ପିକର ଯିଏ ଆମ ଗହଣରେ ରହିବେ ସିଏ ସୁଦୂର ୟୁଏସ୍ଏରୁ ଆମ ସହିତ କନେକ୍ଟ କରୁଛନ୍ତି ତ ମୁଁ ଚାହିଁବି ତାଙ୍କୁ ସ୍ୱାଗତ ଜଣାଇବା Our first plenary speaker for the day is Professor Srinivas Rao Mentor Reddy, and he will be talking on low temperature plasma and emerging green technology for seed priming, improving plant growth and yield, and food safety. We will be welcoming him, but before that, with a round of applause, please welcome Professor B.K. Sahu, esteemed advisor, Sikhya Anusandhana University, Bhubaneswar, as the chairperson. We are having him. And also with us, we will have Dr. Kishore Kumar Behera, the co-chair for the session from the International Center for Agricultural Research in the dry areas. ଗୋଟେ ଜୋରଦାର ତାଳିର ସହିତ ଆମର ବିଶେଷଜ୍ଞଙ୍କୁ ସ୍ୱାଗତ ଜଣାଇବା ଆମ ଗହଣରେ ବର୍ତ୍ତମାନ ଡକ୍ଟର ବେହେରା ଉପସ୍ଥିତ ଅଛନ୍ତି. Let us connect to Professor Srinivas Rao Mentredi, and he is from Alabama A&M University, USA. We'll be talking on low temperature plasma and emerging green technology for seed priming, improving plant growth, yield, and food safety. We welcome you, sir.
Thank you very much. Yeah, I can hear you properly. Can you hear me well?
Yes, sir. You are very much audible. Please go for it. Please share your slides.
Sure. Thank you.
Thank you, sir.
Good morning, everyone. It is indeed my pleasure to participate in this AgriVision 2025. I would like to thank the organizers for giving me this opportunity to present my research at this August conference. It's indeed a privilege to be here and speak about the research we've been doing for about maybe 6, 8 years. And with this, I'd like to start with my first slide. Um, this is a teamwork. I have some graduate students who have done this research. It's an emerging technology, probably 10, 15 years old in the US. And there's some countries like Korea, Japan, um, and Germany is much more advanced than in the US. So this is a new emerging technology. So there are a lot more questions than answers. And I'm going to present research mainly done by my graduate students under my supervision. So we have Sanathanam. He has worked on crop productivity. Dr. Lam Dong, he did a PhD and he worked on sprouting of turmeric using low temperature plasma. Trang Pham also looked at low temperature plasma for enhancing turmeric growth and speed up its process growth cycles so we can grow this in high tunnels. Then Dr. Naichambeng, he's a professor of plant pathology, and he did some low temperature plasma effects on seed-borne diseases, particularly Xanthomonas campestris and stem phylum Botriosum on spinach. And we have Dr. Shu. He's a space engineer, and he's the one who helps us with this new technology, setting up the plasma machine in our lab. So right now, there is a need for innovative solutions. This has been there for a long time. We have agriculture has been innovating itself over the years. So we are now able to break some yield barriers and go on. But there are always challenges. And right now, the biggest challenge we are facing is the climate change effects. And this we are using artificial intelligence, machine learning, drones, precision agriculture, which are transforming the face of farming, making it more sustainable, efficient, and environment friendly. But still, we must keep exploring new technologies that are pollution free, chemical free, they not only improve crop productivity, but also food safety. So they will be having multiple uses and all are geared towards combating climate change. So a major challenge we find is, poor seed germination. A lot of times particularly in climate change effects with the prolonged droughts, erratic rainfall distribution, and the intensities of rains, we often find poor seed germinations leading to patchy establishments, weed growth, and so on, leading to low yields. So we have been looking at this non-thermal cold or low temperature plasma. It's also called atmospheric pressure pulse jet plasma. This is this plasmas could be very hot if they're produced in high vacuum, high temperature conditions. Plasmas can be very hot, running into 300 degrees Celsius. Whereas this low temperature plasma is produced with low voltage and it doesn't affect the plant materials since it is at room temperature or less. So there are many conventional methods for seed priming and improving plant establishment. But then we are looking at new technology that not only promotes seed germination and plant growth, but could also be used for combating plant pathogens. Okay. So there's a lot of research out there proving that cold plasma can be used as a seed primer, either direct or using plasma activated water. We'll be talking some of these things during the part of this presentation. Okay. So what is plasma? It is referred to as the fourth state of matter. It's a weakly ionized noble gas. Noble gas, we're talking about helium or argon. We can also use elemental gases like nitrogen, oxygen, and so on. And these are comprised of positive and negative ions. When we run electricity through this, the gases are broken into the individual species, comes out as a plasma and reacts with the other elements in the air as it comes out of the system. So it has ultraviolet waves also forms hydrogen peroxide compounds as it comes out. So this is generally we subject noble gases like helium or argon and split the gas by passing dielectric barriers. Okay. And these plasmas are now being used pretty expansively in agricultural applications. Okay. So there are different types of plasmas out there. We have man-made plasmas. You see them in manufacturing industries, also in lighting. In medicine, medicine has been plasma was first used as a biomedical tool, and also in space propulsion, fusion energy, and so on. And also in natural, we have natural plasmas out there, particularly when we have lightning, and also Sun-Earth connection, space weather, astrophysical jets powered by black holes, and so on. So plasmas have been always there. This is just a discovery and being harnessed for multiple uses in agriculture science. So this diagram here shows several applications of plasmas in agriculture. And as you can see, um, uh, people have already looked at it for food safety, for microbial decontamination of foods, enhancing crop growth, and also packaging industry, food packaging, and so on. There's a lot of research going on out there. In our case, this is the instrument we use. This is here. We pass a noble gas through this chute here, and then we run the electricity through it, and it breaks up this gas, this purple color, and forms a jet. The jet comes out from here, and this is the plasma, this jet here. And this is very small, maybe few millimeters long. And so there are certain limitations to this because we are using low voltage, so the production is pretty low. But this we have been using pretty effectively. As you can see here, we created using 3D printing a small piece like this brush-like element through which we fitted about 6-8 plasma jets in it. And you can see plasma coming out pretty uniformly. And this is turmeric rhizome. We are treating that to cold plasma. Okay. So Alabama A&M is involved in a big statewide project. So far, we garnered about $40 million for this work. And agriculture is only very small part, about $1 million. But most of the research is in biomedical and industrial applications. About 12 universities involved in this. And at Alabama A&M, I have a team. We have an agronomist, myself, microbiologist, plant pathologist, food microbiologist, food engineer, physicist, and biochemist. It's a good group, and we are looking at plasma in several areas. And this round structure here is again created by using 3D printing, and we are subjecting that to plasma treatment. So the turmeric was first treated like this initially, and they were planted in small flats like this one. And those that were treated, they emerged early. Those that were not treated, they did not emerge, or they came very late. So here you can see the effect on the plant height. The ones that were treated, they're taller. The ones that were not treated were short. Okay. So further graphs here, this first graph here, bar graph, horizontal bar graph shows that the green line shows the sprouting of the early sprouting of treated, plasma treated termic rhizomes, and the red bars are control, that which were not treated. So they took much longer to sprout compared to those that were treated with plasma. Same kind of information here. And you can see the plants, the ones on the left were treated with plasma, and the ones on the right are untreated. And it's variety, Vietnamese turmeric variety 18. So there were differences for their emergence and also percentage sprouting among different varieties. So there are varietal variation also. And in this line graph, we see that this is the plant height, and this is in the greenhouse, and the the these solid lines they show plasma treated. The heights were the plants were taller compared to the broken lines here, which are untreated. And again, some pictures of the plants treated and untreated, and the varietal differences for their percentage difference in the sprouting time to sprout. Okay. So this is an interesting experiment where we had rhizomes that were harvested at 6 months, 7 months, 8 months, and 9 months. And usually 6-month-old rhizomes are very immature and they do not sprout. So we harvested some of them at 6 months, and we treated those with plasma, and half of them were not treated. So in these pictures here, this is control, which is not treated, and these are 6-month-old rhizomes, and the control never sprouted. They just rotted. Whereas the ones that were treated to 60 seconds of exposure to plasma, those to 90 seconds, and then those 120 seconds time of exposure, we can see that at 90 seconds exposure, the plants were much taller. They emerge much faster. So early spotting will give plant the early start, the advantage, so they grow much better. So we have seen this consistently in both 6-month-old and also in 7-month-old. This is a control here. They sprouted very late, 60, 90, and 120. They sprouted much earlier and also had taller plants. Okay. Then we took these plants out and planted in the field. We prepared the field. This is an organic production system. We used poultry manure, cow manure, and vermicompost mix. And we put in these rows and then put plastic. This is the plastic layer here. And then my PhD student planted these plants from the greenhouse. And then we irrigated them using drip irrigation system and also provided soluble fertilizer. And if you see here, the first graph shows the plant height. We didn't see any significant effect here, but still the trend shows that the plants treated plasma, particularly 90 seconds, were taller than at the 60 or the 120 seconds. 60, this aqua blue color, whereas the 120 seconds are yellow. Okay. We saw the same difference here, very consistent in the field. You see the plants here in the middle. This is 90 seconds exposure. Plants were bigger and taller compared to the control 60 seconds and the 120 seconds. So we feel that 90 seconds seems to be uh, optimal for this. So same thing we have seen with 6-month-old and the 7-month-old. And this translated into yield. If you see here, this red bar is 90 seconds treatment. This is 6 months old, 7 months old, 8 months, 9 months, and it went linearly with the age of the rhizomes. And in all cases, consistently 90-second treatment gave higher yield. This is the turmeric that we harvested from each of these treatments. We looked at turmeric, curcumin. Curcumin is the bioactive compound in turmeric. That is what it is mainly sought for by the herbal industry. Excuse me. Uh, Only in one experiment we saw that the curcumin levels were increased when they were treated with plasma. But this was not consistent. We repeated this trial 2-3 times, and we didn't see much effect of plasma on curcumin levels. Okay. That is our research on curcumin. This is a PhD student research, working on papers now. Then we looked at microgreens. Microgreens is a huge industry here. And NASA approached us if we can come with some plants that can grow in in outer space under controlled conditions with less water. We wanted to see if we can induce drought tolerance using plasma and also quick growing. So plants finish their life cycles early, so they'll use less resources. That was the rationale. Generally, for microgreens, this plate one here shows different types of microgreens. They're grown at very high densities, which creates conducive conditions for seed-borne pathogens, plates 3 and 3A and 3B here, a lot of disease, and the whole tray, whole thing will be lost. A lot of, and also sometimes we get patchy germination of the seeds when we put in the trays. So these are a couple of challenges that the microgreen growers face. We thought plasma may be a good remedy for that. So in this picture here, we show this is the source of gas sent here, and plasma is generated, and we tube it into the tube where we put the seeds, okay, and treat the seeds with this. Then we looked at the seed coat control and plasma treated under scanning electron microscope. So the plasma is modifying the seed coat. If you see here, this is control, whereas this is plasma treated. It was a petri dish experiment. We induced drought stress using polyethylene glycol, and saw that there was increase in germination percentage. And then we also looked at the root length, which would indicate drought tolerance ability of the plant. You know, bigger roots, they can explore a larger volume of soil for water and nutrients. So this is the control. These are the root lengths, and this is plasma, the taller roots, longer roots, and also biomass. Generally, in drought tolerance, the plant produces larger root volume to shoot at the expense of shoot. But in this case, we found that there is no such relationship. Biomass also increased as the root biomass increased as well. Okay. So this is a MS student. He's the one who did greenhouse trial. Next, he looked at not only direct treatment with plasma, also you used plasma activated water. We treat the water and then use this water for irrigating these seedlings. And if you see this here, this is the setup. We had artificial light, grow lights, and these are the trays in which we put the petri dishes. And this is the control, much smaller plants compared to those treated with helium plasma for 30 seconds. So there are certain speculations. Like I said, this is a new science. We really do not have a handle on the mode of action, how plasma is increasing seed germination, plant growth. But we believe that certain nitrogen compounds are formed, those called radical nitrogen compounds, and also hydrogen peroxide, and this may be modifying some of the genes, maybe hormone production, certain enzyme production is also being increased. This plate here, some speculation on how plasma may be affecting these changes in seed germination and plant growth. So then we looked at food safety. We looked at we coated spinach seed with fungus called Stemphylum botryoosum. And if you see in this figure here, with plasma treated, the mycelial growth was very much suppressed. These blue bars indicate mycelial growth of plasma treated, and the orange bar is the control. So over time, first 4 days after seed treatment, there wasn't much difference, but 16 days after the culturing, we found that the plasma treated ones had much lesser mycelium. And interestingly, we found that the plasma treated mycelia did not produce conidia. This one here, this black one here, shows the conidia, the reproductive structures through which it disseminates, this fungus disseminates and infects the other plants. That this is in the control, whereas treated with plasma, no such production of reproductive organs. So we looked at SCM. This is a bacteria, Xanthomonas campestris, vesicatoria. It's we treated pepper seeds with this bacterium and treated that with or without plasma. So this plate here shows untreated bacteria. So these are rod-shaped and perfect. There's no change in this. This is a control untreated. Whereas when we treated those with plasma, this is how the bacteria, they kind of swelled up. Some of them broke up. If you see here, the cell wall is broken and probably the bacteria is killed. And also they tended to aggregate more than the untreated. Okay. So we also looked at Listeria monocytogenes on coal smoked salmon. And this line graph here shows as the time of treatment was increased from 0 to 12 minutes, the, the colonies, the populations of Listeria on coal salmon decreased linearly. So this is the plasma instrument here, where we, this is a dielectric barrier plasma. We broke the plasma on the top here and also in the bottom and put the salmon sample in between. So it got treated on both sides. Okay, let me see. Yeah, this is how the salmon is treated with whole plasma. So this ensures uniform coverage of the sample. And then after that, we enumerated the Listeria cytogenes on the salmon, treated and untreated. So we found that somehow it's killing the Listeria on coal salmon. Okay. So what we found was that it improves crop productivity through seed priming, enhance early and faster rates of seed germination, sprouting of rhizomatous crops. We looked at turmeric and probably has the same effect on ginger. We have not done that yet. And also improves plant growth. We have seen the plants growing taller, faster, bigger, more shoots per plant, and also giving higher yields. And also in the case of drought tolerance, we saw that this plasma treated had bigger roots compared to the untreated. And in case of food safety, it kills seed-borne diseases and also foodborne pathogens. So it can be used for as a food safety instrument as well. So we feel that this plasma is a very chemical free and green technology but it needs a lot more research. And we're now trying to get some chemists to work with us to show exactly the mode of action of this. We also be looking at some enzymatic studies using plasma, what kind of changes are happening. So with this project has been funded by a couple of funding agencies. There's a National Science Foundation. Like I said, it's a $40 million there. And then NASA gave us just about $200,000 to do some seed work. So this is a presentation for today, and I'm open to any questions. Thank you. Thank you very much.
Thank you very much, sir, for the wonderful presentation. A round of applause for Professor Mint Reddy. Any questions, anyone?
Yes. Thank you, Professor Srinivasan. Thank you so much for nice presentation about low temperature plasma and how it is being used. But one question that comes to my mind, would you like to know whether it is still in the process of investigation or research, or it has gone to the field for the trials at farmers field, or I mean the large scale use?
No,
Yeah.
this is still, like I said, if you look at the picture, the plasma jet is pretty small, and we are able to do like small quantities, 5 grams of seed at a time. We tried plasma activated water. I have not shown the slides, but plasma activated water also improved antioxidant capacity and also total phenolics in the seeds. But then very small quantities. The problem here is if you want to increase the plasma output, you have to increase the voltage. And if you increase the voltage, the temperature is going up. So the temperature, higher temperatures, the food gets cooked, the seeds get cooked. So we are now doing a lot of trying other ideas on how to scale this up. Um,
Okay. Thank you,
Germany,
sir.
yeah, Germany has done. They were able to treat 1 meter square area of wheat. They made a plasma instrument that they could take it into the field. But in our case, the problem is when you treat water with plasma, the water pH goes down to like 2.5. So we are trying to combat that by adding some kind of a buffers to bring the. So it is still in experimental stage.
Okay, thank you. Then if
So,
anybody has question, then we can take up one question because of time constraint. Thank you.
yeah.
Good morning, sir. Thank you, Professor Srinivas Rao, for an excellent presentation. Myself, Dr. Das, the faculty in Department of Agronomy from OUAT. My question is, am I audible, Professor Rao?
Yeah, yes, I can hear you loud and
Yeah.
clear. Yeah.
My
please go.
question is, when you are priming with low temperature plasma to the seed, particularly when you are growing it, have you ever studied its effect on other soil microbes which are beneficial to soil growth? Its plasma's effect on other soil health conditions.
Yes, right now I have one graduate student. She is working on that. She's just started. We are actually looking at couple of treatments. It's going to be just regular soil. Then we also soil where we amend that with some poultry manure, cow manure, because these all modify the microorganisms in the soil, this poultry manure and cow manure and so on. So this girl, her MS thesis is going to focus on what changes are happening in the soil microbial populations, their species, and also their enumeration. Yeah, that work is, it's not yet started. She just wrote a proposal how she's going to do it. Yeah, we are on it.
Sir. Thank you. Thank you.
Thank you very much.
Over to Chairman.
So thank you, Dr. Rao. Myself, Professor Vijay Kumar Sahu. I'm an agronomist. So really, I'm very happy that the curcumin content is being increased by application of this low temperature plasma. As you know that in our state of Odisha, a lot of people, lot of tribal people, they are doing turmeric, but due to the low curcumin content, they are not getting the proper market price. So really work, it will help all of us.
Yeah,
Thank you, sir.
you're most
Many,
welcome.
many thanks. Yeah.
Yeah, and we are
Thank
open to collaborations with universities. We can come, we can set up the plasma instrument, but costs is a factor. You know, it costs about $35,000-$40,000 to construct one cheap. And if you buy ready-made from Germany, it costs about $150,000. But they are very versatile instruments. Yeah,
Okay,
with all
sir.
the
Thank
sound.
you.
Many,
Thank
many
you.
thanks.
Yeah.
Thank you very much, Professor Mentredi, for your interactive virtual session. Once again, a round of applause for him. Thank you, sir, for joining.
Thank
And
you all
also,
for
as
having
the
us.
session convener, we are having Dr. S.K. Nath, Dr. A.K. Sahu will be the coordinator for the session, and the rapporteurs, Dr. S. Patnaik and Dr. Tapan Kumar Pallai. Moving on for the next session, which is a virtual session by Professor Vaisnava Charan Tripathi from Shraddha University, Noida, New Delhi. Professor Tripathi is with us. Can we please check? Professor Vaisnav Charan Tripathi. Let us move on with the next presentation, which is by Dr. Arundhati Sasmal from OUAT Bhubaneswar. She will be talking on validation of botanical decoction and contortion and microbial constitutia for pest management in crops under natural farming and organic farming. Very warm welcome, ma'am. A round of applause for her.
भाई ओ भौणी माने एठी उपस्थित चासी ओ भाई भौणी मानकु मु एठी स्वागत जनाउची आमे एठी किछि डिस्कशन करिबा जोटा हमर विभिन्न प्रकार पत्र एवं गोमूत्र गोबर एसबुरु तयारी किछि द्रव्य जोथरे हमें रोग पोको को दमन कर परिबा so myself Dr. Arundhati Sasmal I am delivering the speech on validation of botanical decoctions and some microbial consortia for pest management in crops under natural farming and organic farming. At the outset, I want to say that after Green Revolution, the India has transformed from a food deficit to a food surplus country. It is true, we all know it, but do you know the other part of the story or the other part of the coin? That is the second generation problems arised. These are the deterioration of soil health, depletion of groundwater, biodiversity depletion, ecology, environment pollution. Then finally we get the poor nutrition or poisoned food. And the economy is also drastically reduced due to the high input cost. It is high time to take farming from the chemistry lab to connect it to the nature's lab. हमको प्रकृति सांगरे एवं प्रकृति सहित मिसिकिरी हमको ए चाष प्रणाली को आगु को नबाक पड़िबो कारण हमर ए जो जमी चाष जमी एवं समस्तकर स्वास्थ्य मानव कल्याण पई हमको ए प्रकार रो गोटे ट्रांसफॉर्मेशन करबा को पड़िबो The indiscriminate pesticide use has led the several health problems which we know or it is unknown till date. But the issue remains with us. That is the pesticide directly enter to the human body or it may enter indirectly through any other source. That is the high concentration of nitrate in blood. It is reducing the hemoglobin and causing all the nervous, endocrine, reproductive, immune system problems. ଆମର ବିଭିନ୍ନ ପ୍ରକାର ରୋଗ ଆମର ଦେହରେ ଯୋଉଟା ହଉଛି ଏବେ ଆମେ ତାକୁ ଟ୍ରେସ୍ ଆଉଟ୍ କରିକି ଯଦି ଯିବା ତାହେଲେ ଆମେ ଜାଣିବା ଯେ ଆମର ଏଇ ଖାଦ୍ୟ ଯୋଉଟା ଆମକୁ ପୋଷଣ ଦଉଛି ସେଇ ଖାଦ୍ୟରେ ଆମର ଅସୁବିଧା ରହିଛି ତ ଆମେ ସେଟାକୁ ଟ୍ରାନ୍ସଫର୍ମ କରିବାକୁ ପଡିବ contaminated soil, water and vegetation, it is problem, it is creating problem. It can lead to bioaccumulation and poor food safety, nutrition, health, all are in stake. There is decline drastically in the population of the pollinators. Amara mahumachi ebong अन्यान्य उपकारी जीव पोक मानकर संख्या drastically कमी जाउची जोथि के आमे जदि एबे सजाग न होबा ताहाले भविष्यत रे हुए तो आमे आउ एतिकी प्रोडक्शन पाइबानी आमर फल फूल आमे आउ पाइ परिबानी तो ऑर्गेनिक वे और नेचुरल फार्मिंग ऑर्गेनिक फार्मिंग इट इज द वे फॉरवर्ड हमको एथिरे जिबाकू पड़िबो हमको बाध्य एथिरे आमे जिबाकू तो सेथिरे अंडर रे हमर जीरो बजेट नेचुरल फार्मिंग आसुची एसेंशियल प्रिंसिपल्स ता भितरे नेचर्स प्रोसेस एंड एलिमिनेशन ऑफ एक्सटर्नल इनपुट आमे बाहरु किनिकिरी विभिन्न प्रकार कीटनाशक औषध एवं सार इत्यादि को आमे जदि टिके अवॉइड करिकिरी हमर नेचर सांगेरे मिसिकिरी आमे चाष करिबा ताहाले आमे अधिक सफल होबा This natural farming emphasizes on the use of chemical free practice. It is the most important thing. Chemical free practice and easily available farm resource which are manageable for better economy. Economy बहुत important आम पई चासी मानक पई अधिका अर्थ पाइबा को हेले हमर इनपुट कॉस्ट बा प्रथम इनिशियल कॉस्ट को कमेइबा को पड़िबो जदि कॉस्ट कमिबो ताहाले हमें अधिका लाभ पाइबा आमे बहुत खर्च करछे किंतु आमे सेतिकी लाभ पाइ पारु नाही तो हमर गोटे फंडामेंटल प्रॉब्लम डिफरेंस दैट इज द नेचुरल फार्मिंग एंड ऑर्गेनिक फार्मिंग till date we know all these about the organic farming we came from the integrated pest management and all these words it makes us confused actually what is that actually natural farming is the chemical free farming system rooted to Indian tradition actually our India tradition is very rich we should recollect our enriched tradition with modern understanding of ecology resource recycling and on-farm resource optimization. It is largely based on farm biomass recycling. आमे सेही farm रे ही recycle करिबा with major stress on biomass mulching, use of on-farm cow dung, urine formulations, maintaining soil aeration, exclusion of all synthetic chemical inputs. आमे जेते परिबा आमे chemical input किनिबा को avoid करिबा आमे हमर farm रे पाउथिबा गोबर गोमूत्र एवं सेहितिर विभिन्न प्रकार मल्चिंग आच्छादन ए सबुर व्यवहार करिबा ऑर्गेनिक फार्मिंग कोन दिस एक्सक्लूड्स केमिकल फर्टिलाइजर्स इन द सेम वे एज वी थिंक इन द नेचुरल फार्मिंग बट हियर इज द ऑप्शन फॉर परचेसिंग bio inputs like the biopesticides biofertilizers we can purchase from the market and we can use it just a replacement of chemicals by the biologicals. This is the theme or this is the concept of organic farming. But in the other way, natural farming emphasizes on the inputs which are available in the farm itself, in and around the farmer. चासी पाखरे सामनारे पछरे जेतिकी जिनिस अछि हमें सेइथिरु मध्य चाष करि परिबा हमर चाष भलो भी हबो भलो अमल भी हमें पाई परिबा एटा होचे हमर नेचुरल फार्मिंग एवं ऑर्गेनिक फार्मिंग मोर और लेस सेम बट जस्ट डिफरेंट स्कूल ऑफ थॉट्स एम्स एंड ऑब्जेक्टिव ऑफ नेचुरल फार्मिंग हमर जदी चिंता करिबा ताहले हमर परिवेश सुरक्षा एटा होचे मोस्ट इंपोर्टेंट नेचुरल फ्लोरा फॉना प्रिजर्वेशन रिस्टोर सॉइल हेल्थ डाइवर्सिटी ऑफ प्रोडक्शन प्रमोट naturally available beneficial insects बहुत उपकारी कीट अछंती बहुत उपकारी जिंस अछंती आमे तारो संरक्षण करिबा तार आमे परिचालन करबा प्रमोशन ऑफ लोकल ब्रीड जोटा कि लुप्त हो जाउची हमर आस्ते आस्ते देसी जिनिस we will promote all this desi because they are acclimatized to the environment physically amara ei environment रे amari desi जीव जंतु माने से माने रही चंती तांकर रोग कम से मानकर आउटपुट अधिका यूज ऑफ नेचुरल एंड लोकल रिसोर्स बेस्ड इनपुट्स हेल्दी न्यूट्रिशनल एग्रीकल्चर प्रोडक्शन प्रोड्यूस फॉर कंजप्शन इकोनॉमिक्स सबुठु अधिका हला पैसा इकोनॉमी टा डेवलप करची तो हमें कौन से व्हाट टाइप ऑफ थिंग्स वी कैन यूज दैट इज बीजामृत जीवामृत आच्छादन एंड फास्पा बीजामृत is a seed treatment chemical biological formulation for seed treatment. Jivamrut is a soil enriching consortium or rich in microbial consortium. Then achhadan or mulching, it is main thing is to reduce the weed, and second thing it improves the soil by adding some biomass. Then phosphor means some vapor inside the soil so that all the root growth amar gachar chera ebang amar jo upakari jiba ye mane achanti jibanu achanti semane bhalare badi paribe to ei sabu ame nei kiri ame natural farming ero components ku nei ki ame chaliba scientific understanding of the biological system. So for us, like our educationist or the scientist, the problem is the issue is to validate or to say whether it is working, how it is working, why it is working, all these things. So creating values beyond the yield. आमे सबु बेले हमरा yield किंबा हमरा अमल को लक्ष्य रखी चली परिबनी. We have to think beyond it. We have to think for the society, for the environment. So किछ हमरो decoction we are using. What is that decoction? Decoction means the liquid extract obtained by boiling plant materials in water. सीधा कथा रे आमे जदि पानी रे सिझेई करी किछि बाहर करचे ताकू हमरो रोग पोक समस्या पई आमे व्यवहार करचे सेटा को कुहा जाय डिकॉक्शन जेमते आमे निंबरु बाहर करि परिबा ब्रह्मास्त्र एटसेट्रा कंकॉक्शन just different is that it is the mixture of the natural ingredients. Particularly, the concoctions offer incorporate different plant extract, microbial solution, natural elements. आमे ताकू किछि मिसै कि रखिबा fermentation हबो एवं fermented product कोन products made from the fermentation of different botanicals and along with the animal dung and urine. So cow urine is rich in germicidal, antibiotic, antimicrobial and medicinal properties. आमे स्त्री हमरो ओडिसा रो प्रत्येक घरे घरे हमरो लेडीज माने जो गोमूत्र गोबर रे घर चारिपटे ये करंती दैट इज एटा को हमरो भाइरस किंबा बैक्टीरिया रो एंट्री ता भीतर हेई पारेनी रोग जीवाणु आसी पारेनी एटा हमरो चिराचरित प्रथा वी आर आवर रिचुअल in this context some research has been done at OET to validate all these formulations, whether it is really efficient, whether this is really working against the insect pest diseases, etc. So one experiment was conducted at Regional Research and Technology Transfer Station Bhubaneswar OET in paddy variety Pratiksha during Kharif seasons of 21-22. 21-22 मसीह ए परीक्षा कराइथिला. Here Bijamrut, Jivamrut, neem oil, cow urine mixed with neem oil, then some neemastra, brahmastra परीक्षा कराइथिला. ताकू देखा हेतिला हमरो जो औषध आमे व्यवहार करचे दुकान रु किनी के आनी क्लोरोपाइरिफस. You know all the farmers भाई भोनी मान समस्त जानि चंती क्लोरोपाइरिफस अति परिचित तो आमे ता सहित कंपेयर करिके देखिथिलु. We compared it to know the bioefficacy. Bijamrut, Jivamrut, amara normal protocol, standard protocol along to Niti Ayog, जो थिरे अच्छे हमारा सेथिरे हमें से प्रोटोकॉल को फॉलो करिकी हमें गोबर गोमूत्र चूर्ण माटी एवं पानी रे एई प्रोपोर्शन रे मिसई की हमें तैयारी करथिलु. निमास्त्र एवं ब्रह्मास्त्रटा सिझेई की तैयारी हेतिला. निमास्त्र मिसई की सेटा दिस इज ए concoction. So from this experiment we found that the plants treated with Bijamrut, soil application with Jivamrut, and the application of Brahmastra solution, it gives very good result with the stem borer, then leaf folder, then brown plant hopper, population reduction. माने आमर धान रे जो मुख्य पोक कीट समस्या देखा जाए आमे देखिलु जे एथिरे बहुत भल भाबरे सेटा कंट्रोल रे रही पारुची हमर आर्थिक देहली सीमा बा ईटीएल तळे रहूची इट इज गिविंग at par yield, it is statistically at par yield with the chemical. And जदि आमे पैसा हिसाब करिबा इंक्रीमेंटल कॉस्ट बेनिफिट रेशियो केते खर्च कले केते पाइले केते अधिका खर्च कले केते अधिका पाइले एई जदि आमे हिसाब करिबा ताहाले हमर खर्च निश्चय हमर बोटानिकल आमे जोटा सिझेई गिरी पकेइचे सेतर तो किछि पैसा खर्च होइनी सो वी गेट बेटर बेनिफिट दैट इज द 4.71 in comparison to 2.87 ICBR in the chemical because we purchased the chemical from the market. These are the preparations. So finally, the seed treatment with Bijamrit, soil application with Jivamrit at 30, 45, 60 and 75 DAT, foliar spray of Brahmastra at 30, 45, 60 and 75 DAT found to be most effective in managing the major insect pests by reducing 58%, 64%, 70% in stem borer, leaf folder, BPH population with yield improvement and economic. So, এই ପ୍ରଣାଳୀଟା ଆମେ ଚାଷୀ ଭାଇ ଭଉଣୀ ମାନଙ୍କ ପାଖକୁ ଆମେ ପ୍ରସାରିତ କରିପାରିବା ଆମେ ଏଭଳିଆ ଭାବରେ ଚାଷ କରିପାରିବା we can get the higher benefit. Then ଆଉ ଗୋଟେ ପରୀକ୍ଷା କରାହେଇଥିଲା ତ 2019-20 ରେ organic ଆମେ ଯୋଟା ବାସୁମତି ଆମର local organic local ବାସ୍ନା ଚାଉଳ लीलावती गोटे निया ହେଇଥିଲା ଆମର ଭଦ୍ରକ ର ସେଟା ଭେରାଇଟି ସେଥିରେ ଆମର total complete package of practices it was assessed and these package of practices includes the seed treatment then dhanicha incorporation FYM application Jivamrut soil drenching neem cake application pheromone trap and the release of Trichogramma chilonis egg parasitoid then spray of the neem oil or that is the Beauveria bassiana and neem seed kernel extract and alternate of neem oil and Beauveria bassiana. This experiment was done to get a complete package of practice for growing the local basmati rice instead of the chemical or the ame binna chemical le kemiti basna dhano kari pariba se bishay re amara experiment heitila. All the visitors also came to the field to see the plot and these some analysis was also done for the Jivamrit and Bijamrit in the Department of Soil Chemistry, Microbiology and here we found that the seed treatment with Pseudomonas fluorescens, Dhanicha incorporation, vermicompost, neem cake, then pheromone trap, Trichogramma chilonis release and Jivamrut application along with the spray of Beauveria bassiana जोटा कि हमको एंटोमोपैथोजेनिक फंगस हमर कीट नियंत्रण पे व्यवहार करा जाए तो एई समष्टि रे एई जो पैकेज टा हमें देखिलु दैट गिव्स अस पेस्ट रिडक्शन पोको कम हैला एवं अधिक हमको लाभ मिलिथिला. So this package of practices it was extended to the farmer society through KVK. Seed treatment with Pseudomonas, green manuring, then neem cake, Jivamrut, Trichogramma chilonis, Beauveria bassiana spray. It reduced the pest population and yield improvement. Then some student research under my guidance, my supervision has already completed in the Department of Entomology on this evaluation of organic management modules against major insect pests in paddy. The objectives are like this: organic management of the pest. Second one is the impact on the beneficial insects. What is the impact? Because we know that the chemicals are having very bad impact or very negative impact on the natural enemies. So our other side, we have to assess that these botanicals, ei jo ame ବ୍ୟବହାର କରୁଛେ botanicals तार उपकारी कीट उपरे कोन प्रभाव रहिछि एटा कोन ता पई खराब एवं महुमाछी पई भी खराब की हमें से सब को भी टेस्ट करथिलु बायो आसे करथिलु हमर लैबोरेटरी भितरे जे प्रकृत रे पोक मरुची कि से खाउनी ताकू देखिबा पई so these are the treatments that was the experiment was conducted by the student and these are the laboratory test. Yes, this is the result. Efficacy of different management modules against insect pest in paddy. We have seen that this foliar application of Brahmastra along with alternate spray of neem oil and Brahmastra along with the application of the Jibamrut, it gives highest reduction. सबुठु अधिका कांड बिंधा पोको सेथिरे दमन हेतिला एवं सेथिरे हमर जो धळा केंडा हुए जेतळु धान पाचे सेतिक बेले सेटा मध्य 49.56% कम हेतिला तो ऑलमोस्ट 50% रिडक्शन आमे यदि ग्रॉस चासी
Madam, you have 2 minutes more, madam.
Okay, thank you.
Please.
So these are the reduction in the pest population and this is the impact on the beneficial insect. We have seen that almost all the practices are very good, very beneficial to the beneficial insects and improving the yield. This is the yield improvement of 20% and it is the laboratory test 20% of the solution 20% of the solution of Brahmastra, it was found very much effective against the leaf folder in the laboratory condition also. So this Nimastra and Brahmastra can be a weapon for managing the pests. It has some limitations because these are botanicals, you know. यार किछि अपकारिता भी रही जेमति कि एटा बहुत शीघ्र नष्ट हो जाए तो सेठमे हमको बारंबार स्प्रे करिया को पड़े. So in this context we have validated to some extent and the work is continuing at the Odisha University of Agriculture and Technology. Finally, I want to say that organic farming and natural farming practices are low input demanding, energy efficient and holistic production system which play a crucial role in sustainability of agriculture production. So we must go ahead with this, with nature. Thank you.
Thank you very much, madam, for the wonderful presentation. आमे गोटे क्वेश्चन आम चासी भाई मानक ठारो नबा जदि रोग पोक दमन को नहीं मैडम को आपण किछि प्रश्न करबा को चाहुचंती प्लीज पचारंतु. यस प्लीज.
मते जी कहबे कि आपण जिंदे लास्ट साइड मेंशन कले यार बहुत जल्दी नष्ट हिज जने सिंगल जने फार्मर ता पाखरे एते मैन पावर न थांती तो हमें थरे करि देले ए सॉल्यूशन टाको केते दिन पर्यंत हमें पकै परिबा. सेकंड थिंग
6 months.
6
प्रथम
months
क्वेश्चन रो आंसर होची तारो आमे सेल्फ लाइफ इंग्लिश रे सेटा को कहै छै सेल्फ लाइफ आ आमे जदि ओडिया रे कहबा ताको आमे केते दिन संरक्षण करी रखि परिबा. ए सॉल्यूशन टा तयारी करिवार 6 मास पर्यंत सेटा आमे बॉटल रे काच बोतल रे काच जगह रे बंद करिकि रखि परिबा एवं तारो जो सेल्फ लाइफ बढ़ेबा पई मध्ये हमर एठी एक्सपेरिमेंट एवं रिसर्च वर्क वी आर डूइंग.
अच्छा सेकंड थिंग मतलब जी जेमते एटा गोटे नेचुरल जिंस रो तयारी होची यार रेसिड्यू भी रहत आमे जेतले केमिकल फर्टिलाइजर आनुचे जो पिठी रे पके कि जो स्प्रे करछ मशीन रे सेतरे पूरा सॉल्यूशन टा पूरा किछि रेसिड्यू रहउनी फ्लो रे सेटा स्प्रे करि पड़छ एथरे सेही जिंस पॉसिबल कारण नेचुरल जिंस तो रेसिड्यू रहइबो तो स्प्रे होबा नै.
स्प्रे करिबा पूर्वरु गोटे कपड़ा कन्ना जोटा हमर सूतार कन्ना पर्टिकुलरली साड़ी किंबा धोती ए टाइप रो जिंस रे ताकू छाणीबा को पड़े हेले सेटा स्प्रे करा हबो तापर ता पय नोजल भी टिके चेंज करके बड़ होल वाला यूज करबे.
धन्यवाद.
हां आउ सेटा भलो स्प्रे हबा पय सेथरे जदी 1% डिटर्जेंट माने टिके हाथ रे पिंच साबुन गुंड मिसाई देबे तो सेटा होची तार सरफेस फ्लोएबिलिटी बढ़ाई दबो एवं अधिका भलो भाबरे स्प्रे हे परबो.
यस मैडम एठी रहिबे तो आपण मैडम को साथ आलोचना करि परिबे.
टिके मोर गोटे क्वेश्चन.
यस मैडम.
मैडम क्रॉप इंसेक्ट गुडा को से भी इंसेक्ट आ बेनिफिशियल इंसेक्ट भी इंसेक्ट ताले तांकर किछि फिजियोलॉजिकल डिफरेंस अछि कि जादर कि बोटानिकल्स रे ଆପଣଙ୍କର କ୍ରପ୍ ପେଷ୍ଟ ଗୁଡାକ ଲିଥାଲ ହେଇପାରୁଛନ୍ତି କିନ୍ତୁ ବେନିଫିସିଆଲ ଇଫେକ୍ଟ କାହିଁକି ଲିଥାଲି ହେବନି?
ଏଥିପାଇଁ ଲିଥାଲ ମେନଲି ହଉଛି ଯେ ଆମର ଯେତେବେଳେ ବୋଟାନିକାଲ୍ସ ଆମେ ପକଉଛେ ସେଟା ହଉଛି ଯୋଉ ଲିଫ୍ ର ସରଫେସ୍ ରେ ସେଟା ରହୁଛି that is acting as a anti-fidant or feeding deterrent to the pest. କାହିଁକି ନା ପତ୍ରକୁ ସିଏ ଖାଉଛି କିନ୍ତୁ ନ୍ୟାଚୁରାଲ ଏନିମି ପତ୍ର ଖାଉନି। ନ୍ୟାଚୁରାଲ ଏନିମି ଯୋଉଟା ଉପକାରୀ ପୋକ ସିଏ ହଉଛି ତାର ଅଣ୍ଡା ମାନେ ଆମର ଯୋଉଟା ଅପକାରୀ ପୋକର ଅଣ୍ଡା କୁ ନଷ୍ଟ କରୁଛି କିମ୍ବା ତାକୁ ତା ଭିତରେ ସେ ପଶିକିରି ଅଣ୍ଡା ଦଉଛି କିମ୍ବା ସେ ତାକୁ ଇଏ ଖାଉଛି ତେଣୁ ଇନଡାଇରେକ୍ଟ ୱେ ଏଟା ଡାଇରେକ୍ଟଲି ପତ୍ରକୁ କିଛି ଖାଉନି
ନାଇଁ ମୁଁ କହୁଛି
ଯୋଉଥିପାଇଁ ତାର
ବେନିଫିସିଆଲ ଇନସେକ୍ଟ
କହିଲି ପରା sir, beneficial insect is not directly eating the, it is not eating the leaf or the, it is not associated with the leaf area. So it is indirect ରେ ରହୁଛି ସିଏ କାହା ଉପରେ ଡିପେଣ୍ଡ କରୁଛି ସିଏ ହଉଛି ପେଷ୍ଟ ଉପରେ ଡିପେଣ୍ଡ କରୁଛି।
ହନି ବି ତ ପୁଣି ଖାଉଛି ନା?
ହନି ବି ଟା ହଉଛି ଯେତେବେଳକୁ ଆମର ଏକ୍ଜାକ୍ଟଲି ସ୍ପ୍ରେ ହଉଛି ସେଇ ସମୟରେ ଯଦି ହନି ବି ଆସୁଛି ତାହେଲେ ହୁଏତ ତାକୁ ଟିକେ
ଏଇ
ସେଇ
botanical
ପ୍ଲଟ୍ କୁ ମାନେ ସେଠି ନ ଆସିକି ସେ ଆଉ ଟିକେ ସାଇଡ୍ କୁ ପଳେଇବ ବଟ୍ ଇଟ୍ ହାସ୍ नो ରେସିଡୁଆଲ ଏଫେକ୍ଟ ତାପରେ ସେ ଆସିବ
Thank you, madam.
ସେ ଡାଇରେକ୍ଟଲି ତାର କଣ୍ଟାକ୍ଟରେ ଆସୁନି।
Okay, thank you, madam. Thank you.
I think ରାଘବ ଭାଇ ଏଠି ଅଛନ୍ତି, ଅରୁନ୍ଧତୀ ମ୍ୟାଡାମ ବି ଦିଦି ବି ଏଠି ଅଛନ୍ତି, କିନ୍ତୁ ଅସୁବିଧା ହବନି କିଛି ପରେ ବୁଝିବା ପାଇଁ। Let us go to next session. Thank you.
थैंक यू वेरी मच मैडम आम टिके जल्दी सारिबा को चेष्टा करिबा कारण आज जेहेतु शेष दिन वैलिडिक्टरी सेरेमनी मध्य रही जहाँ समय सुधा करने को पड़ो। वर्तमान प्लीज स्वागत करिबा डॉक्टर सोपन के त्रिपाठी फ्रॉम OUAT भुवनेश्वर सर इज टॉकिंग ऑन एक्सप्लोरिंग आर्सेनिक टॉलरेंट एंड एक्सक्लूजन डोनर्स फॉर यूज इन राइस ब्रीडिंग। अ राउंड ऑफ अप्लॉज फॉर सर एवरीवन प्लीज। LED screen please. Okay. Again.
Thank you, AgriVision, to give me opportunity to present my paper. My topic is exploring arsenic tolerant and exclusion donors for use in rice breeding. I am fascinated to take this topic because arsenic is a silent killer, and particularly in rice, I would talk about that. So malnutrition due to heavy metal is a chronic problem, as rice is actually is a staple food for more than half of the world population. Arsenic toxicity is a serious problem, particularly in rice, because we are using most of the time the groundwater for irrigation purpose, which is actually contaminated with arsenic. Arsenic binds with soft tissues in our body, and it causes particularly cancer and also cardiovascular problems, renal problems, respiratory problems, and many other problems associated with our body. Arsenic is a toxic heavy metal. It is a toxic heavy metal, and it also making a life-threatening problem to our body. Significantly, it also, besides that, it will also reduce the crop yield significantly, and therefore this topic must be a relevant one. ଆମର ପାଖାପାଖି 200 ଆର୍ସେନିକ୍ କମ୍ପାଉଣ୍ଡ ପାଖାପାଖି ଆମର ଆର୍ଥରେ ମିଳେ। ସୋ ୟା ଭିତରୁ ଆମର AS3 AS5 जोटा कि ना इनऑर्गेनिक फॉर्म जोटा कि ना सबुठू हमरो बेसी टॉक्सिक बोली जना अछि। Inorganic forms arsenic, it is actually more toxic to our plant system because of the fact that it is having a low LD50 value, that means 15 to 40 mg, whereas in case of arsenic 5, it is actually having LD50 value ranging from 220 to 800. Indica rice actually it accumulates more arsenic than that of the Japonica rice, and therefore in our indica varieties whatever we are cultivating, जोटा हमर धान हमर कल्टिवेट करते सेतरे बेसी आर्सेनिक अमाउंट बेसी एक्युमुलेशन हुए। तो सेति भाई हमर जोटा ग्रेन हमें खाउचे कि धान चाउल खाउचे सेतरे बेसी आर्सेनिक आसुची कंपेयर टू जापोनिका राइस। Therefore, identifying the most suitable arsenic tolerant genotypes with least accumulation of arsenic in grains under lethal condition of A stress seems to be top priority in our research. Methodology. So I have taken 131 rice germplasm and it was tested in TRD with 3 replicates. Arsenic stress was given in form of sodium arsenite. LD50 was actually assessed over a concentration of 0, 5, 10, 15 to 20 ppm. For stress tolerance, we have maintained 5.4 pH maintained kora jaychi 5.4. Also the nutrient solution we have taken using 15 ppm. And we have advocated also, we have offered also controlled environmental condition to experiment this particular strategy. Experimental observation. So far as experimental observation is concerned, LD50 LD50 for different rice varieties has been calculated, and they have been categorized into tolerant, highly tolerant, then moderate tolerant, moderately susceptible, and susceptible to arsenic toxicity. Stress tolerance index has been also calculated based on the fruit and also based on the root length. Seedling vigor as well as the relative seedling vigor was also calculated. And lastly, leaf area damage was taken into consideration to score the arsenic tolerance or arsenic sensitivity. This table says that over a concentration gradient of 0, 5, 10, 15, 20, different varieties we can calculate the status of arsenic tolerance based on this LD50, and the varieties which are exceeding 15%, 15 ppm LD50 value, those are categorized as tolerant. You can see the growth behavior. It is the growth of the plant is decreasing from 0 to 20 ppm. So this is the mass screening of rice varieties for arsenic tolerance. You can see some of the plants, they look green. Those are actually tolerant to arsenic toxicity. So we have categorized these few varieties which are marked as tolerant based on the LD50, and besides that, we have also calculated stress tolerance index based on root length and root length, as well as relative vigor index, and also LAD means leaf area damage. These are the varieties which have been screened out as tolerant versus sensitive varieties. You can look how they behave in the screen. So this is a pictorial diagram for data points of STI, that means stress tolerance index. Stress tolerance index based on root and fruit, and seedling vigor index, and also leaf area damage. The scale indicates the peak towards periphery. It is giving much more tolerance to the particular variety, whereas in case of LAD, the peak towards the center, it gives a impression that it is much tolerant than that of the other variety. So far as correlation among the different seedling characters are concerned, germination percentage, it gives a negative correlation with that of the LAD, that means leaf area damage. Whereas relative seedling vigor index, and also stress tolerance index, they are highly connected. That means these traits we can take into consideration. One of the traits we can take into consideration for screening of arsenic tolerance. Here I have given two pictures, left and right, two plotted pictures. So left one is control and right one is under arsenic stress. That has been treated under 15 ppm up to seed maturity. That means up to completion of the life cycle. And I have tested the arsenic content of root, leaf sheath, leaf blade, husk, and kernel. And it was seen that the arsenic accumulation is much higher in tolerant variety, particularly in root, compared to that of the root in case of susceptible variety. But the thing is that whenever it is assessed in leaf sheath, leaf blade, husk, and kernel, the amount of accumulation of arsenic accumulation तार leaf sheath, leaf blade, husk और grain रे आस्ते आस्ते कमी जाउची compared to that of the susceptible variety. So this gives a impression that there is a clear-cut partitioning of arsenic whenever we are go to the top area of the plant. That means in the grain there is much less accumulation in the tolerant variety, whereas in the grain much more accumulation of the tolerant variety of the susceptible variety due to higher uptake of arsenic to the grain directly. Here is a screen diagram. So here it indicates the three principal components which are giving much more contribution towards the phenotypic variation. So that we can take up the principal component analysis in the next slide. So this principal component analysis, it gives a impression that C7, C6, C5, that means seedling vigor index, then relative seedling vigor index, then stress tolerance index, and also seed germination. These are the characters which are highly connected. That means one of the traits we can take into consideration for arsenic tolerance, and these are having higher loading towards the positive side of the principal component 1, whereas C8, that means LD, that means leaf area damage, it is giving much more negative value, negative loading in the principal component analysis. This gives an impression that the second quadrant, that means left side 2 quadrant, they are the varieties which are designated as sensitive variety, whereas the fourth quadrant, that means lower right quadrant, it is the varieties which are having, which particularly some of the varieties which I have already indicated earlier, those are indicated here. Those are categorized as highly tolerant. I have also taken attempt to have a clustering pattern of those varieties, and it has been seen in a circular diagram where all the varieties have been clustered. All the tolerant varieties have been clustered together in the red circle. So this gives a impression that we can have a simple methodology without having low financial cost towards experimentation to identify a few arsenic tolerant varieties to go forward for rice breeding program, and we can safeguard the food security and nutritional security as well. Thank you all.
Thank you very much, sir. Any questions, anyone? किछि प्रश्न जदि आपण मानकर अछि सर को आपण माने पचारि परिबे? Yes, गोटे प्रश्न आमे पचारिबा सर किछि समय आम सहित अछंती आपण तांक सहित आलोचना करि परिबे, बट वर्तमान पई जदि किछि प्रश्न आपणक मन रे अछि पचारि परिबे.
Sir, one question, sir. Sir, आमे जो पॉपुलर राइस वेराइटीज गुडा सब करूचंते बे कोटा बेसी आर्सेनिक accumulation करूची तार किछि आपण भाई रिसर्च डाटा अछि कि सर? पॉपुलर राइस वेराइटी सपोज पूजा करूचंती, ललाट करूचंती, प्रतीक्षा करूचंती, सरला एई भितर कोटार बेसी आर्सेनिक
Directly susceptible. They are actually the high transport of arsenic.
Okay, thank you, sir.
But we are not getting arsenic toxicity because of the fact that in many areas we are not having arsenic. But the mining areas which are actually having much more arsenic, if we will put them in those areas, they will accumulate much more of arsenic in the grain, and we will be succumb to that particular dreadful disease, arsenic toxicity.
Okay, sir, thank you.
Thank you very much, sir. A round of applause for Dr. Tripathi, everyone. बहुत बहुत धन्यवाद सर. चाषी भाई मानक सरल भाषा रे मध्य बुझेइचंती. वर्तमान ଆମେ ଆମର ପରବର୍ତ୍ତୀ ବିଶେଷଜ୍ଞ ଯେ ଅଛନ୍ତି ତାଙ୍କ ସହିତ କନେକ୍ଟ କରିବା ସେ ଆମ ସହିତ ଭର୍ଚୁଆଲି ରହିବେ our invited speaker Dr. Deo Mishra from Bayer Bioscience Hyderabad. He will be talking on crop health management for sustainable rice farming. It's welcome, sir.
Yeah.
Let's
good
get
morning.
connected to Dr. Deo Mishra. Sir, am I audible?
Yeah, I can hear. Hello.
Sir, can you hear me?
Yeah, I can hear.
Sir, please
Hello.
unmute yourself. Please unmute yourself.
Unmuted already. Hello.
Sir, you are not audible. Please unmute yourself.
Hello.
Yes,
Can you
sir.
hear
Now
me?
you are audible. Please share your screen. Please
Yeah.
welcome Dr. Deo Mishra with a round of applause, everyone.
Thank you. Thank you, madam. Hope you are able to see my screen.
No, sir, not yet. Please share your screen.
Is it visible now? No?
No, no, sir.
Let me share again.
Yes.
Is it visible now? It should be now visible.
Yes, sir. Now it is visible. Make it in presentation mode.
Sure.
Yes, sir. Now it is visible. Please go for it.
Yeah, thanks. Thanks, madam. And good morning, everyone. So I'm Deo Mishra, part of Asia breeding in Bayer Crop Science based out at Hyderabad. So I'd like to thank the organizer for inviting in this conference for giving this talk. So my talk will be on the crop health management for sustainable rice farming. So just to let you know some information about me. So I'm working as a Asia Plant Health Risk Assessment lead in Bayer. I completed my education from Pandnagar University and have been working in company since last almost 22 years on multiple aspects related to plant breeding, pathology, entomology, grain quality, and some of the activity that is required for the breeding support like genetics, tissue culture, and all those. So looking into the mega trends that is really driving the need of innovation into agriculture. So we see a lot of challenges in modern agriculture as of now. And that is linked with the growing population. And that is what is expected, like adding more than 2 billion people by 2050. And that will give pressure on the agriculture system because that growing population will require more than 50% more food and feed by that particular time. And then what is happening, because with the growing more food, there is also a lot of pressure coming from the climate change. And what we are expecting, around 17% loss will happen with the changing in the climate, and also the kind of pressure that is going to put on the ecosystem. But on top of that, this change in climate will also lead into the increasing pressure from the disease insect and weeds. So that is something we need to really keep in mind when we start advancing in agriculture system to protect the crop from growing pressure of the diseases and insects. And in that sense, we need to really start looking all those three components that is really required for a disease or insect intensification. So like environment, pathogen, and host, we know, and then customer, how they are tackling all these. So that is something, the challenges is phased, and understanding all these components will really help us going forward to means sustainably manage the crop pests. Going on directly on the topic. So rice, as we all know, it is the important crop, and in true sense, rice is life for almost half of the world population. And looking into the trend that is going now, so we see that there would be a lot of pressure on the rice as well. So as I told, so rice I'm not going to explain this one because everybody know the importance of the rice. So looking into the mega trends in the rice, so we are seeing that there will be a lot of losses that will be coming from the diseases, insects, and weeds. So whatever we are producing or whatever the potential of rice production is there as of now, so more than 37% is being lost every year by diseases, insects, and weeds. So that is the major challenge that we need to address. And when we talk on the plant health, specifically, so when we analyze all those data since last multiple years, so we found that bacterial leaf blight, brown plant hopper, insect, and rice blast. So these three are the major contributor in those loss that is happening all around the world. And I'll just go through to touch some of the innovations that has been done at our side, specifically for these major three traits that is affecting the rice crop health. Like first one is the bacterial leaf blight caused by Xanthomonas oryzae, Pathopara oryzae. So we try to understand what kind of variability the pathogen is bringing so that we can have good resistance deployed in the product that is being deployed in the farming community at farmer level. So if you see here on this table, so after analyzing more than 1400 pathogen isolate, we could find that there are high level of variability in the pathogen, and then we need to have multiple resistance source when we are targeting the resistance product against this disease. So this study help us to decide that what are the resistance genetics that you will see in the left-hand side of this table will be important to deploy when we are targeting the resistance against this disease. And the pathogen is really variable, so it is spread across the geography where rice is being grown. And there is a lot of variability in the pathogen population. That's what you will see in this map in the left-hand side of the slide. So like all these areas, they are like dominated by major pathogen races. So we need to really have the genetics that is strong enough to contain all these strains which are present in the geography when we are going to launch a product, whether it is variety or hybrid. Another innovation, we were looking into the genomics of the pathogen so that we can start looking how it is evolving and whether the deployed genetics will be effective when it is being deployed in all those geography as we have seen. So this study has been done in collaboration with the CCMB and CSI IMTech in Chandigarh and also with the California State University to pinpoint the region, genomic region in pathogen that is responsible for breaking the resistance that is being deployed, for example, here XA21 as a major resistant genetic. So we really made good progress, and now we are in position to track the pathogen population movement based on its genomic information. Based on all those information, we were able to deploy the resistant genetics, and you will see here in the picture right-hand side, the product that has been deployed, it is working well across the Asia and providing big relief to farming community against this disease like this bacterial leaf blight. Second one is the brown plant hopper. It is also very dynamic insect and it can destroy the crop when insect population is increasing. And few years before, if I remember correctly, in 2017 and 18, there was epidemic specifically in Odisha region and a lot of damage, I believe, has been done during that time. So here also we studied the population variability in the insect, the population that has been collected from different regions in the country, and then try to identify some of the resistant genetics that can be really effective against all these population variability. And then based on the resistant genetics, we deployed that in some of our product, and you will see here if resistant genetics is deployed, how it behaves. So under no like the segregation of resistance and susceptible here, so you will see insects, they prefer to land and multiply on the susceptible genetics, and they were not able to multiply on the resistant genetics when they were planted side by side. So that is how it is showing the promise that the native resistance is able to protect the crop against this dreaded insect as well. And we have seen that kind of differentiation. Like, for example, here in the chart, you will see this blue bar is indicating susceptible genetics and green is the resistant genetics. And we have seen the insect multiplication on both kind of genetics. And you will see resistance is multiplying very easily on the susceptible genetics, but on resistance, its multiplication rate is very slow. So it means the resistance is really hindering the feeding, egg laying, hatching, and all other insect related those parameters, so that it is keeping the population at very low, so that it is not able to cause the damage on the plant. So this is what we have done as of now on these plant health traits. So we started our journey sometime in 2008, and by now we have worked on these 3 major traits, and we have now product deployed in the countries, specifically in Asia, and they are really performing well and serving the farming community to save their crop from these dreaded diseases. And this slide is just to show that what kind of impact these traits are making, specifically the bacterial blight, brown plant hopper, and another one, abiotic stress, the submergence, that we have also worked and deployed. So as of now, by 2024, all these products are being grown in more than 4 million acre area. And that way they are really saving a lot on the farming fields in terms of the yield loss that could have happened because of these insect or disease infestation. And also kind of some of the challenges that farmers feel, they really look, for example, for bacterial diseases, they apply a lot of antibiotics, and that is not safe for the human health also. So in that way, it is really saving a lot. So if you see the additional cost, like $5 to $10 per acre is being saved by the resistant genetics that has been deployed in case of bacterial blight. And also in case of BPH, we are able to save almost $30 per acre because of this resistant genetics. So this is something, the way that we are trying to really help the farming community by way of managing the crop health in rice. And you can see one of the farmer from Chhattisgarh, she was really looked from that angle, because she has grown some of those products, and she has been given this recognition by Honorable Prime Minister Narendra Modi for growing these products and getting a lot of yield on that one. Another thing that I want to highlight here, like the cropping system shift that is happening now, because there is a lot of push that is coming from the government and also International Rice Research Institute, and even from the institutes in India, those are working on rice, on shifting from the transplanted rice to direct seeded rice, because it is going to save a lot of resources in terms of the labor, in terms of the water, in terms of the methane emission, and supporting the climate in terms of the being more sustainable. So this is also bringing some kind of challenges also when we talk on the crop health. And if you look in this picture in the left-hand side, so DSR will bring these many benefits as I just discussed. But there would be some challenges looking into the plant health, specifically the increase in the soil-borne pathogen like nematodes and all those, and a lot of micronutrient deficiency that may come. So some of these diseases, as we observed since last 2-3 years, they will be in growing trend as compared to transplanted, although you will see that in transplanted system also they are there, but some of these like root knot nematode, so that is going to increase in direct seeded rice. So in future, we are really going to work, and we need to work collaboratively on some of these traits. Those are hard to manage, so that this new system of growing rice becomes more sustainable and more profitable for the farming community. So this is the change that we are going to look. But in the future, we are also looking, because only one way of managing the plant health is not going to be effective. For example, if we talk only on the genetics or native trait. So we are looking into the prediction as well as the chemistry along with the genetics, so that it becomes more tailored approach and more beneficial, and there will not be much pressure on the insect or pathogen to evolve frequently. And this is how we are seeing that there is really a great progress that is happening, and that is really helping to all our farming community to get more yield means per unit area. And one of the example here, I would just say that how tailoring two approaches is beneficial in managing the bacteria, this brown plant hopper. This picture is from Odisha, and you can see after 6 spray of pesticide like this insecticide, even farmer were not able to save their crop. But with the genetics and 2 spray, the crop was saved, and they were able to harvest a good amount of the yield there. And second example is also from Chhattisgarh, and where with 1 spray, farmer was able to harvest the healthy crop. And that is how we are looking. It is not only like eliminating chemistry and depending on the genetics only. So it is like a kind of harmonized approach where all the available things we can combine and go into integrated approach as more tailored and more holistic and more sustainable. So
Excuse
this
me,
way,
Dr. Misra.
yeah.
Can you please sum up? You have for 1 minute more time, and
This is
kindly please, yeah.
last
Thank you.
slide. So this is how we are just looking into that, how we are going to progress. So based on disease simulation and pest quantification, we are going to recommend that what kind of genetics and pesticide that should be integrated, and then also based on the precision application, so we can have tailored pest solution going forward, and also having better crop yield and healthy crop. So that is all from my side. So it was not the like one man work. So it was done in collaboration with multiple institutes. You can see here, and we have really come up with very great partnership and great results. So thanks everyone for listening and giving me time to present what we are doing here. So if there are any query or questions, so I'm
Yeah, we can take one question because of time constraint, if it is there from the audience.
okay, sure.
Any questions, anyone? Okay, thank you. Thank you very much, sir, for the wonderful presentation. Thank you for joining. Moving on now, let us
Thank you.
welcome our
Thanks, ma'am.
invited speaker from Indian Statistical Institute, Kolkata, Dr. Soren Das. And his topic for the day is multifaceted eco-physiological behaviors towards salt management in mangrove plants. Please welcome, sir. जेहेतु आज वैलिडिक्टरी सेशन मध्य रही अनेक गुड़े स्पीकर मध्य अछंती तांकर प्रेजेंटेशन पाई तो आम ये टी ब्रेक नौ ना किंतु बाहर टी जाइए जब मैंने चाह सेक्शन जो रही आपड़ जब आने चाहते प्लीज गो एंड हैव आपन जाइपारे प्लीज हैव सम टी किंतु आम कौन ब्रेक बर्तमान नबा ना ओवर टू डॉक्टर दास
Good morning, everybody. I'm talking about the multifaceted ecophysiological behavior towards salt management in mangrove. the mangrove plants are equally commercially and biodiversity important to our mankind, as because it the protect the vast coastal area which is most fragile rather. Mangrove plants widely distributed over 120 tropical and subtropical zone of the world, and it is according to 2020 14.8 million hectare covered by this area. This equatorial zone of the world covers the mangrove, as because equatorial zone receives maximum sunshine and temperature and humid also. Mangrove requires that thing. The most extensive mangrove area is found in South and Southeast Asia. That means that the largest mangrove swamps in a single block situated in the Indo-Bangladeshian zone, that is in Sundarban. In Sundarban, the richest mangrove biodiversity are there. At least moreover 40 to 42 plant species are there. We don't get anywhere such a huge number of plants diversity like Sundarban area. Between 2000 and 2020, 677,000 hectares of mangrove were lost. That is our unfortunate, which was partially offset by the expansion of 339,000 hectares of mangrove, resulting more or less 284,000 of hectares in the 20 years. The highlight the dynamic nature of mangroves, the coastal ecosystem transitioning from one prompt to another as an environmental condition and change. Mangroves are always both economically and ecologically important to us, as because in the production view, mangrove ecosystem service worth of US dollar 33,000 to 57,000 per hectare per annum. Apart from this economical point of view, the climatic regulation is much more important. Mangrove forest, the dense greenery considered as the lung of the nearest urban settlement. Apart from this, the tourism, weather condition, coastal protection, it is the main aspects of mangrove. During the past tsunami or like sea surges, the affected area are much more where mangroves were destroyed, but where mangroves are in intact condition, their destruction rate is much lesser. This is the ecosystem service of the mangrove, but there are some threat also. Which are the threat? Threat from mankind and threat from the ecological condition. From mankind, all that we are progressing and encroaching the mangrove land by ourselves, putting the settlement or other tourism for aquaculture, for agricultural land, expansion of agricultural land. Moreover, unplanned exploitation of the mangrove forest. That is a big issue to the environmentalists. If the mangrove forest abolishes, what will do? The periodic sea surges always will be much more devastating than present condition. This is the one thing, and soil erosion will be controlled by this mangrove ecosystem. So now I'm talking about the abiotic stress. Plant stress are two types, biotic or abiotic. Abiotic stress that means environmental stress, soil nutrition, water logging, much more salinity also. Plant stress is defined as any change in environmental condition that produce less than ideal plant expression. Now, effect of salt stress. forget about stress. Effect of salt management in mangroves. There are two type of salt management should have that plants. One is ionic stress, another is osmotic stress. Osmotic stress, you know that the water is much more salinity, that is water potential is much more high. Mangrove plants itself have to maintain their water potentiality in the cell much more higher than the sea water. Otherwise, water conduction will not be there. If not, deficit of cell water potential and then stomatal closure, ultimately it is affects the photosynthesis. And second thing is ionic stress. What is ionic stress? How to maintain the potential target pressure of the cell? Mangrove always try to accumulate within the cell some compatible solute. What are the compatible solute? Not on the inorganic solute. If they accumulate inorganic solute within the cell sap, then the other metabolic processes will be hampered. That's why they always stored in the cell some protein, some compatible solute, some amino acids, like these are make them the more target pressure within the cell sap, by which the water conduction will up. Interference of the normal photosynthesis generate much more histo ROS or RNS. That is, I'll come back. And breakdown of antioxidant defense system. Ultimately, senescence of the cell. This is the general response of the cell salt management, not stress. Mangroves normally grow in the salty region. That is why I am not agree to take them as a salt stress environment. They are salt loving. Now the parameter addressed here: net photosynthetic rate, chlorophyll content, mesophyll conductance, specific leaf area, stomatal conductance, photosynthetic nitrogen use efficiency, free amino acids, and antioxidant systems. These are the common formula by which the photosynthesis can be calculated, and stomatal conductance and the specific leaf area like this. I don't go in the detail. The average rate of net photosynthesis was higher in fresh water. We compared 5 mangrove element in plants in their normal in situ condition or then salty condition and ex situ condition in mesophytic condition. This comparison results something interesting, that Bruguiera gymnorrhiza, Exocaria agalocha, and Phoenix peludosa, those are dominating mangroves in Sundarban area, but Heritiera fomis and Xylocarpus granatum. I repeat, Heritiera fomis. It is named, vernacular name is Sundari. By name of the Sundarban is determined. Or otherwise, once upon a time, when salinity somehow reduced lesser, in Sundarban, there is plenty of Heritiera. But nowadays, you go, you have to search for the Heritiera. It is almost extinct, red bee state plant in ICUN. That is why the point of interest of mine. In saline and non-saline condition, the net photosynthesis is lesser in saline condition and higher in non-saline condition. Why? See, in the open forest, there are maximum amount of power they absorb. PAR, photosynthetically active radiation. In normal process, excess power accumulation leads to excess photosynthesis. Not that. Excess power accumulation leads to high leaf temperature. If leaf temperature comes to the threshold value, then the photosynthesis will close. That's why in forest region, the photosynthesis cannot in a steady state condition. It is up and down. When the leaf temperature rises more, as because of excess power, the stomatal conductance will be lower and stomatal will be closer. Then photosynthesis close. After some time, it again start. That's why in the normal in situ condition, that means in saline condition, the photosynthetic behavior is rather apart from the normal mesophytic condition. Stomatal conductance. Stomatal conductance depend on the accumulation of the solute within the stomatal cell, by which the considerable decrease of stomatal conductance was noticed under salt stress condition. Why? In salt stress condition, they are all not stressed. I repeat that it is not stressed. In salt management condition, they accumulate much more solute within the cell sap as well as within the stomatal cell. That's why stomatal conductance is somehow decreases in the in situ condition. That means in Sundarban condition. That's why stomatal closures are easier. And chlorophyll content in Bruguiera gymnorrhiza, Exocaria agalocha, and Heritiera fomis, chlorophyll content in leaf was higher under non-saline condition. In Heritiera, an insignificant increase of 0.7% was estimated in saline reduced in soil. In Phoenix paludosa and Xylocarpus granatum, elevated soil salinity increased the total chlorophyll content by 22 or 7% respectively. Third is specific leaf area. Specific leaf area determines the photosynthetic efficiency of a plant. In all the 5 species, SLA was lower in higher salinity. That means in situ condition, the mangrove plants have some lower ability to get photosynthetically active function. And mesophyll conductance, one is another thing. Mesophyll conductance determined by the ratio of total mesophyll tissue divided by total leaf thickness. This determines the leaf target pressure within the leaf, by which the water conduction will go. Next, in amino acid, it is described as equivalent to percent leucine. The major free amino acids are aspartic acid, alanine, proline, tryptophan, tyrosine, phenylalanine. Aspartic acid and phenylalanine and alanine were detected from all the taxa, but considerably lower amount was detected from the plants grow in non-saline environment. Free proline was detected Bruguiera, Phoenix, and Xylocarpus. There where it is absent in Heritiera. That means Heritiera physiologically to some weaker than the other dominating mangrove. That's why Heritiera means, I again repeat, that Sundari plant. It is somehow physiologically as well as anatomically, I found it is weaker than the other major growing mangrove plants in the Sundarban region. Day by day, salinity increases in the Sundarban region due to global warming and sea water rising. Second thing is how much sweet water influx within this delta matters. If we see that Bangladesh part of the Sundarban is somehow virgin. Why? Bangladesh part receives Ganga and Brahmaputra, sweet water from Ganga and Brahmaputra much more better than the only Ganga through the Indian part. That's why Indian part of the Sundarban salinity is certain rising. The antioxidant enzyme mainly I covered in 2, peroxidase and SOD. As salinity increased, considerable interspecific differences was observed in isoforms pattern of peroxidase and SOD. Different isoforms of these 2 enzymes increases either with their number of occurrence or the magnitude.
Sir, excuse us. Can you please take 1 minute more and sum up here kindly? Yeah, thank you.
No problem. Now, the important thing I conclude, but if the antioxidant system somehow break in the cell, if the ROS or RNS content increases in the cell, if unattended that ROS or RNS by the antioxidant system, peroxidase, SOD, phenolic compounds, they are the scavenger of this RNS and ROS. If this is the balancing factor to withstand the plant itself. If ROS and RNS is much more higher and much accumulating within the cell sap, then plant will be senescence. So, if salinity rises, this increase of ROS and RNS happening. No, thank you.
Thank you, Dr. Das, for excellent presentation. One thing I'd like to know from you, because you are an expert in this line, how best you can manage mangrove forest, particularly in their Odisha, Bhitarkanika, and your is their Sundarban, and that is nowadays in a great danger to the climate change. It is vanishing day by day, and the area is reducing. So this must, because the forest department is also not taking a interest for management. Even the government is not taking proper management. So you suggest something, some points, how best you can manage, and that should be go to the government as a proceedings. Thank you.
This is the main purpose of study. We have to maintain the wastewater surplus. That means that can be done by the using of the river. In Bangladesh region, there are two rivers, mainly one Ganga and one. Both combined wastewater reflux is that one is much higher than the same river from the eastern side of the western side of the
Yeah, another point regarding the waterlogging, you also mentioned, because that should be proper drainage facilities also be provided in those mangrove areas. Yeah. Different, yes. Okay.
Such a way, each plant is to be in the next
Okay. Time is short. We'll discuss in the lunch time. So thank you, Dr. Das, for the excellent presentation on the mangroves. Thank you. So next, we have Dr. Raghunath Satpathy, virtual. ठीक है, देख ले. Dr. Raghunath Satpathy from GM University, Sambalpur.
Yeah, am I audible, sir?
Yes, sir.
Okay. Let me share my screen. Is it visible, sir?
Not visible.
So please share it.
Yes, sharing. Some issue. Issue, I think. There is a issue.
Sir, you can go back and again share your slide. Once go back and again share your slides. So is it audible to you? So are you there? Disconnected. ଆଉ କିଏ ଅଛନ୍ତି virtually? କୁଲଦୀପ ତ୍ରିପାଠୀ ଅଛନ୍ତି? କୁଲଦୀପ ତ୍ରିପାଠୀ ଆଚ୍ଛା ଏଇଠି ରହିବେ ଓକେ ଅବୁବାକର ସାଲିହୁ ଅଛନ୍ତି ସେ? ଅବୁବାକର ସାଲିହୁ ଅଛନ୍ତି? ଆଚ୍ଛା ଏହମଦ ଅଲିୟୁ ଏହମଦ
Yeah,
ଅଲିୟୁ
Abubakar
ଜଣେ ଥିବେ
Salihu. Excuse me, Abubakar Salihu, I'm around.
ଏପଟଟା ଅନମ୍ୟୁଟ୍ କରି ଏପଟଟା ମ୍ୟୁଟ୍ କରିଦିଅ
Hello. Can you hear me,
ଟିକେ ଟିକେ
please?
ଅନମ୍ୟୁଟ୍ କର ଟିକେ ଗୋଟେ ସେକେଣ୍ଡ yes sir we can hear you please be connected we will get back to you again ଆଉ ଅନମ୍ୟୁଟ୍ କରିଦିଅ ଏପଟଟା
Is it my turn to present?
Next invited speaker, Dr. Raghunath Satpathy from GM University, Sambalpur. Today's topic is prediction of phytochemicals against plant pathogens, a computational analysis. Sir, now can you hear me? Epata unmute achana. Dr. Raghunath Satpathy, we welcome you to AgriVision 2025. Is it audible to you? Sir, ଶୁଣି ପାରୁଛନ୍ତି ଆପଣ? ଅଛନ୍ତି? ଏପଟଟା ମ୍ୟୁଟ୍ ନାହିଁ ତ?
Am I able?
Yes, sir, you are very much audible. Please welcome Dr. Raghunath Satpathy, everyone. Let us have a round of applause for him. Over to you, sir. Please share your slides now.
Yeah, I'm just sharing. There are
Yes.
some huge with system.
LED screen, please.
Not able to share. There are some problems, not able to share this.
Okay, sir, you can resolve the issue meanwhile. Please be connected. You can resolve the issue meanwhile. Let us now get connected to.
Okay, I think he can give an extempo and so that summarize the topic within the timeline. That is also possible if the presentation is not working.
Yes, sir, our chairperson is speaking to you. Is it audible to you, sir?
Yeah.
Sir, if you are not able to share your slides, you can
Yeah.
summarize your topic
Yeah, that I will do.
if it is possible.
Yeah.
Yeah, Dr. Satpathy, if possible, suppose it is not working this presentation on the slide,
Yes, sir.
then
Sir.
could I please request you to give an extempo and talk on your slide few lines and summarize it within
Yes.
10 minutes.
Yeah.
Thank you.
Yeah. But just a minute. Okay.
Press 9. Screen share करना
Yes, Dr. Tripathi, please.
Dr. Satpathy.
Satpathy, yeah, sorry.
Doing the problem.
सर आपनकर स्क्रीन टा शेयर हेनी जदी पॉसिबल आपनकर स्क्रीन टा शेयर करंतु
प्रॉब्लम होची
आज्ञा आज्ञा सर सर आपण टिके आपनकर जो स्टेयरिंग इशू रही छै टिके रिजॉल्व करि परिबे meanwhile we can take another presentation if it is okay with you. Is that
Actually,
okay with you, sir?
Sir,
You
actually,
can take some
mostly-
time and you can take some time and
I have some other assignments, so that's why I want to.
okay, then please try to.
Satpathy sir.
Again.
Hello. ଆପଣ ତାହେଲେ ଆପଣଙ୍କର ରିସର୍ଚ୍ଚ ବା ଯୋଉ ଟପିକ ଅଛି ତାପରେ ଆପଣ ଟିକେ କହି ଦଉନାହାନ୍ତି 5-10 ମିନିଟରେ ଆମକୁ so that audience ସେଟା ଶୁଣି ପାରିବେ.
So we can hear you. ଆପଣଙ୍କର voice ଶୁଣା ଯାଉଛି. ଆପଣ ଆପଣଙ୍କ slides on କରନ୍ତୁ. ଆପଣ ସେଇଠୁ go through କରିକି ଆମକୁ ମାନେ କହି ପାରିବେ ଆପଣଙ୍କ research work ବିଷୟରେ.
Okay.
Again, please, sir. Should we start, sir?
Yeah.
Yes, sir. Please go for it.
So, good morning all. So, I will be talking about prediction of phytochemicals against plant pathogens, a computational approach. The work basically deals with the molecular modeling work to find out an alternative phytochemicals against the rice blast disease. So rice blast disease is basically it is caused by Pericularia oryzae. So what we did basically, we have identified one target in these particular things. So we are targeting a particular protein, essential protein in this particular pathogen. So for that, we have we downloaded all these proteins of this rice plant from the all the proteome from the UniProt database, as well as we also retrieved all this protein sequence of this Pericularia oryzae pathogen. Then we did a local blast search of these pathogenic proteins against the rice proteome. Then we identified there are 116 number of proteins which are unique to this pathogen, which are not available in the rice genome. So we then manually curated those 116 number of proteins and identified there is a protein called kineurinase 3.7.1.3 EC, which is basically is considered as the target of this particular pathogen. So we predicted the structure of this particular protein by using the AlphaFold surfer. Then we validated the structure by using Ramachandran plot and other allied softwares. Further, we searched the Chinese traditional medicine database for a suitable phytochemicals. For that, we use a server known as drug rep server. In that particular server, it identifies it actually predicts the pockets in the particular protein, and it actually finds the phytochemicals which can fit to this particular binding site of this protein. So in this aspect, we selected 100 phytochemicals for that. Then all these 100 possible phytochemicals or promising phytochemicals were screened by using docking score, by using log p-value, by using its molecular weight, and toxicity parameters. So all these screening were done by using Swiss ADME server as well as ProTox server. So after that, we identified only 2 phytochemicals which can be the suitable for this particular enzyme, which can be used as the inhibitor for this enzyme. Again, after docking, post-docking analysis we did, we identified there is a phytochemical known as allyl A, which the basic source is Alisma plantago, which binds tightly to this particular protein, which is the result is comparable to the natural ligand kineurin of this particular enzyme, but is used in the metabolism of the tryptophan metabolism. So this allyl A binding was again validated by using the molecular dynamic simulation by the Yasara structure model. So this by using this molecular dynamic simulation for 50 nanosecond, we again analyzed several parameters like RMSD, RMSF value. Then there is a radius of gyration. There is number of hydrogen bond dynamics. Then we also identified what is the ligand movement RMSD analysis. Also, we did the molecular mechanics Poisson-Boltzmann surface area MMPBSA analysis to find out the binding energy. So all this analysis indicated that this the complex of this protein and the we identified this allyl A is the binding complex is very much stable. So we can predict that the allyl A phytochemical can be used as a suitable inhibitor for this particular pathogen. So that is our thing. But however, we need to validate those things by using the experimental work. So this is the summary of all these things. So if you have anything, you may ask.
हेलो
Hello.
थैंक यू डॉक्टर सतपति कहार कुछ
Yeah.
क्वेश्चन है जदि सारंग पे कि पचारिबे हमें क्वेश्चन नहीं परिबा one or two
Actually,
questions
results I could not show you. There is issue with this particular thing.
okay so still then we have some questions from audience and please
Yeah.
Thank you, sir. Sir, you told that you have studied on 2 phytochemicals. Allyl A is one, another one, sir?
Another one is I just forgot the name. It is there in the slide.
Okay.
Another phytochemical was there. Both are comparable
Yes.
things.
So is the whether someone is better in comparison to other or some nothing like
Yes,
that?
yes. I told you that after immediately things we screened by several parameters, but then the main parameter what we have taken is the hydrogen bonding.
Yes.
The other phytochemical, as I told, it do not have any hydrogen bonding, but the standard molecule or standard ligand and this phytochemical allyl A, the two I got there are 4 to 5 hydrogen bonds. That's why
Okay.
we have selected allyl A for that
Okay.
purpose.
Thank you. Thank you, sir.
Thank you very much, sir. Any questions? Anyone? Any? ଆଉ କେହି ପ୍ରଶ୍ନ କିଛି ପଚାରିବାକୁ ଚାହିଁବେ? Anyone else? Thank you. Thank you very much, Dr. Raghunath Satpathy, for presenting
Thank you.
your topic. Thank you very much for joining, sir.
Thank you.
Next up, we have our invited speaker, Dr. Kuldeep Tripathi from ICAR NBPGR, New Delhi. He is he there? Sir, are you here? Okay, moving on, Dr. Abubakar Salihu. Is he there? Okay, let's get connected to him.
Yes, I am there. Good morning to you all.
Yes, a very warm welcome to you, sir. Dr. Abubakar Salihu is from Modibo Adama University, Yola, Nigeria. Sir, please share your screen. He is talking on gender analysis of access to agricultural production technologies among farming households in eco-local government area, Nigeria, Jombe State.
Yeah.
Sir,
So-
is it audible to you?
Yes, yeah, it
Yes.
is. It's on.
Yes, sir, it is visible. Please share your screen, slides.
All right. Just a minute. I'm trying to share the screen.
Sir, it is not visible now. Share your slides, make it in presentation mode.
Yeah, I'm trying to share it, but I think some issues because I'm using a tab to do the presentation.
Sir, can you speak from little distance? You are not audible properly.
Am I audible now?
Yes, yes. Yes, sir.
Okay. All right. I'm trying to share it. I think maybe some little issues. I'm using my tab for that purpose.
Right now, we can see your name. Please share your slides.
All right. So can you see it? I have shared it.
No, sir, not
Is
yet.
it not yet? I think I'm having some issues with the sharing also. If it's possible, let me present the findings in a summary form in some few minutes.
Sir, your slides are not visible yet.
Yes, I said I'm having some issues. I'm using my tab because I have some issues with my computers. I'm using the tab to share the screen and I'm having some problem while sharing it. So if it's possible, can I summarize my findings in some minutes?
So you can go for it.
All right.
Yes, sir.
The
Sir, once
slide-
try to show the slides.
Yeah, right now I'm having issues with sharing the scripts, but then I can present a summary verbally, just like what the
So
other
you
speakers
can
did.
try to share your slides. If not, then you can give an extempo.
All right. The title of the research article is Gender Analysis of Access to Agricultural Production Technologies Among Farming Households in Akko Local Government Area, Nigeria. So the research consists of making findings using primary data. Data were collected from farmers. We tried to make it comparative between male and female farmers to see the different types of technologies that each have access. The research paper aims to address three objectives: to describe the socio-economic characteristics of the female and male farmers in the study area, which is Akko local government area, then to identify the agricultural technologies accessible to female and male farmers, and then to analyze gender differences in accessing the agricultural production technologies by the respondents. So the study was conducted in Akko local government area. Akko
Sir,
is a
sorry for the
district-
interruption. 1 second, sir. Please hold on. Can you turn on your video if it is possible?
All right.
Please turn
Just
on
a
your
moment.
video.
All right.
Yes,
All
sir.
right.
Now, if possible, please share your slides, share your screen.
Screen sharing.
Now we can see you, sir. Please share your screen once, slide screens. Icon below, you can try from there.
Yeah, I tried now showing not allowed. Let's see.
There is an icon below. You can try from there.
Yes. Yeah, I saw the icon, but still. This is the icon share. Screen is not sharing.
Okay, sir. Now your video is on. You are visible. Please continue from there. Please continue.
All right.
Yes,
It's
thank you,
on.
sir.
Yeah, so like I said, the study was conducted in Akko local government area. Akko is just like a district. Here in India, you call it district, but in my country, we call it local government area. Farmers were selected, male and female farmers were selected across different villages of Akko local government area. So we collected primary data from the farmers using structured questionnaire on the objectives I have highlighted. Okay. So in total, we selected 172 male and then 76 female farmers. However, not all were used for the analysis. So the tools for analysis used in analyzing the data collected were descriptive statistics. We use just frequencies and percentage to describe the socioeconomic variables of the concerned farmers. Then again, proportional two-sample t-test was used to compare gender differences in access to agricultural production technologies. We tried to see among the male and female farmers, which of the groups have more access to agricultural production technologies? And is the difference between the two in terms of access significant or not? So here is a summary of the socioeconomic variables, information we got from the farmers, okay? So all in all, male and female lumped together in terms of age. In terms of age, the highest proportion of the farmers, which accounted for around 30.0%, okay, constitute of farmers within the age group of 31 to 40. In terms of marital status, 70.3% of the respondents were all married. In terms of family size, just like the average tradition of African countries, we usually keep large family size. So more than half of the respondents have a family size of between 4 to 6 persons. On the average, 4 to 6 persons in a household. Then in terms of educational level, the results revealed a low literacy rate amongst the farmers, with only 16.5% of them attaining tertiary education, while the majority of them, which is around 43.8%, completed secondary schools. In terms of farm ownership, that is how the farmers acquired their farmland. Most of them, precisely around 54.6%, inherited their farmlands. So this is all with socioeconomic characteristics. Then now, in terms of the agricultural production technologies accessible by male and female farmers. So here we categorize the technologies based on two decisions. Decision either a technology is accessible or not accessible. So in terms of how we arrived at whether a technology is accessible or not, we listed the different types of technologies available in the study area, and then developed some sort of scale with 4 options. And we gave values of 1, 2, 3, and 4. So if you add 1 plus 2 plus 3 plus 4, it will give you 10. Now, if you take the average, it's 2.5. So we allowed the respondents to freely tick the level of access they have to each of the technologies. At the end of the day, we estimated a cutoff mark, which is the summation of the points. That is 1, 2 plus 3 plus 4, which is 10 divided by 4 is 2.5. So each and every farmer that has a score of 2.5 and above, we deemed that technology as accessible to him. A score less than that indicates inaccessibility. So the technologies were categorized into insecticide and application methods. There are different application methods, that is aerial application, foliar application, and then and somehow. So based on that, among all those that we found, the insecticide aerial application was not accessible to male farmers, but it was accessible to female farmers. Then in terms of labor equipment, that is access to labor equipment, we have planters, knapsack sprayer, and then other labor equipment. That was the category we used. So knapsack sprayers were accessible to all male and female accessible, but planters was only accessible to male. The female doesn't have access to it. Then we look at the access to technologies through mobile apps. Okay, in terms of the way we look at the agri app smart farming, this app was not accessible to both male and female farmers. This could be attributed to maybe low literacy level among the farmers. Then we have what we call the My Crop Manager app. This was accessible to only male farmers, but female farmers do not have access to it. And then more other mobile application apps, it was accessible to male farmers, but female farmers do not have access to it. Then we look at accessibility to different types of fertilizer, getting the required quantity that they want. We look at NPK 15, 10, 20, NPK 15, 15. So these two categories of fertilizer were accessible to both male and female farmers. Then the application method for the fertilizer, the broadcasting method, was used by male and female farmers, did not use it. Then we look at accessibility to processing equipment. The maize sheller machine was accessible to maize farmers, but it wasn't accessible to female farmers. The millet thresher was accessible to male, but not accessible to female farmers. Then we now look at different improved seeds. We look at the groundnut, Psalm 36, we look at Psalm 37, and then others. So we look at about 8 improved seed varieties. Amongst the 8 improved seed varieties that we look at, constituted of mainly groundnut, cassava, cowpea, and sorghum, only 5 were accessible by male farmers, while to female farmers, it was only 3 that was accessible. Now, the last aspect of the presentation, we look at the gender differences in access to agricultural production technologies. So here, we listed the different types of, broad types of technologies, which constituted insecticide and application method, herbicide and application method, labor equipment, processing equipment, and so on. So here we look at the proportion of male and then the proportion of female that have access to each of the technology. And then we use the proportional sample t-test to compare between the two. So in all the technologies that we have compared, all the groups of technologies, we discovered that male farmers have more access to the technologies compared to female farmers. And then the statistical difference was quite significant as revealed by the peer sample t-test. So based on this, the research concluded that males farmers, the research concluded males have more access to technology compared to female farmers. Notwithstanding, even the access to to the technologies by male farmers needs to be improved as depicted by the results. So some recommendations were made. Farmers, regardless of gender, to explore diverse opportunities and provide them with training and resources to enhance socio-economic outcomes. Also, accessibility of agricultural production technologies for both male and female farmers in the study area by increasing availability of the technologies, awareness campaign, providing financial assistance, and strengthening extension services should be looked into. Also, access to agricultural production technologies by implementing gender-responsive interventions that specifically address the unique needs of male and female farmers should be taken care of. So as a result of this study, we submitted a proposal to the Gombe State government. So currently now they have launched a particular program whereby farmers are now trained, both male and female farmers are trained on different technologies. And then among them, some were identified and then trained as community seed entrepreneurs. The aim of it is to train them to be able to produce their own improved seeds, which can be marketed and sold within the community so that other farmers within the community can have more access to it. That has been going on, I think, for a year and a half. And then remarkably, this year we conducted an assessment and then found out that there was a remarkable improvement in their yield compared to the previous years. So this is a summary of what I have. Thank you very much for listening. Thank
Okay.
you. I'm done
Thank
with
you.
the
Thank you very
presentation.
much, sir, for the wonderful presentation. A round of applause for Dr. Abubakar Salihu, everyone. Any questions? Any questions, anyone? Jadi kichi prashna achi pachari paribe apana mane. Sir, please stay with us for a while. Any questions, anyone? If you want to ask any question to Dr. Abubakar Salihu, please go ahead. Sir, please, you can ask one question if you want to. Any remarks? Any remarks, sir?
Yeah, well, I started without giving an introduction of myself. I'm Dr. Abubakar Salihu, a senior lecturer with Modebo Adama University, Yola. I did my BSc and MSc degrees from Baro University, Kano, and then proceeded to India, to the famous Banaras Hindu University, India, where I obtained my PhD in agricultural economics in 2019. And then since then, I have been working as a lecturer in Modebo Adama University, Yola. So I thank you very much for giving me this opportunity.
Hello.
I consider India as my second home. However, due to late issuance of visa, currently I'm in Delhi, and due to logistic problem, I couldn't proceed down to OUAT Bhubaneswar. I intended coming, but it wasn't possible. My visa was released very, very late. So thank you very much. I appreciate it.
Thank you very much, sir. That was absolutely a commendable presentation. Could we please have a louder round of applause for Dr. Abubakar Salihu? He was from Modibo Adama University, Yola, Nigeria, and his topic was gender analysis of access to agricultural production technologies among farming households in eco-local government area, Jombe State, Nigeria. Thank you very much, sir. Once again, a round of applause for him. Moving on, now let us get connected to Dr. Ahmed Aliyu Santureki from Modibo Adama University, Yola, Nigeria. Sir is joining us all the way along from Nigeria. Could we welcome him with a huge round of applause? And he is presenting on effect of different concentrations of neem seed oil in management of cucumber mosaic virus of pepper, that is Capsicum annum 1, under greenhouse condition. Let's get connected to Dr. Ahmad Aliyu Santuraki. Could you please have a round of applause for him? जोरदार तालीर सहित तांको स्वागत जणाइबा आमे नाइजेरिया रु आम सहित शामिल होउछंती डॉक्टर अहमद अलियू संतुराकी. So, acha, you will be. Ma'am, your slide is paused. Am I audible, ma'am? Please unmute yourself, ma'am. Your voice is not audible. Please unmute yourself if I am audible to you.
Okay, am I audible enough now?
Yes, yes, yes.
Okay. So,
please welcome
I'm-
Dr. Santuraki, everyone. Ma'am, please share your slides.
All right. All right, so good morning, everyone from our Nigerian time. I'm representing Dr. Santuraki Ahmed Aliyu, who is working on effects of different concentration of neem seed oil in the management of cucumber mosaic virus disease. So my name is Victoria Moses, and I'll be representing him as he will not be able to be part of this meeting. So pepper is an herbaceous plant belonging to the family Solanaceae, which also includes important vegetables such as tomatoes, potatoes, and eggplants. And we have about 5 most common species of pepper, which is common for human consumption. And some of the species are listed down here. So the world production of pepper is about 6.7 million tons of fresh pepper and 4.14 million tons of dried pepper. And Nigeria is one of the highest producer of pepper in Africa. And pepper is grown majorly in the northern part of Nigeria, and a lot of cultivars exist in northeast, and we have a lot of cultivars which names are written here. So I'm showing us a slide. These are pictures of different pepper cultivars we have. We have the Dan Mayo, Dan Fufore, Sabondogo, and Dan Bembu, and Dan Kano. So there are a lot of economic importance of pepper for food security, which is one of the importance of this conference. Poverty alleviation, as it's a source of income to a lot of farmers who practice the cultivation of pepper in Nigeria. It's also a source of job creation in this part of the country. And it also helps in food palatability, because there are a lot of food that you can consume. I don't know if it's the same thing over with the other side, but here in Nigeria, there are a lot of food you can consume without pepper. So which shows the importance of this plant. So it's very rich in vitamin A, beta carotene, very rich in vitamin A and vitamin C. It's rich in fiber, antioxidant, and a lot of economic importance. So I would like to move straight to my materials and metal. This this experiment was carried out in the greenhouse at Modibo Adama University in the year 2021. And Yola is a state in is a state in Nigeria, which is located in the Guinea Savannah ecological zone of Nigeria. And the temperature ranges from 20 to 45 degrees Celsius with a relative humidity of 55 to 65%. it's quite very hot over there. So the pepper seeds were sourced locally from local markets, from local stores, and same to the neem seed oil, which has Azadirachta indica, was sourced also locally from local stores here in Adama, and we sourced the the CMV inoculum from infected plants through symptoms, obviously, and before the soil sterilization took place, because we had to sterilize the soil from other pathogens that could be present in it, and this was done for 4 hours and the soil was left to rest for about 7 hours before we started work on it. So nursery was made, and the nursery were made in the greenhouse and were transplanted after 3 weeks, and we planted after 3 weeks in this greenhouse, and inoculation of the isolated inoculum was added to the healthy plants, was inoculated to the healthy plants at 7 to 10 days after transplanting. So, and treatments was applied, about 2 weeks after that. So the next slide is showing. Okay, so the design that was used was completely randomized design, and we had about 5 treatments. T1, which is the control. T2, which is a ml of neem oil plus 10 millimeters of water. T3 is 2 milliliters of the neem oil with 10 liters of water, which, as you can see from my screen, showing all the different treatments that were used during this experiment. So the results show that pepper plants inoculated with CMV showed disease symptoms at 1 week after inoculation. The analysis of variance for the incidence of cucumber mosaic virus indicates no significant difference at week 1 and 2, showing significant difference on the 3rd to 4th week after inoculation. And I have a table next I'll be showing us. Okay, so most times when we carry out an experiment like this, we take data on incidence, data on severity, data on plant growth parameters, and data on it. So this is the data on incidence, which is showing that there was incidence, a very high incidence on the control of about 42.8%, which is significantly high. And when we were using 4 ml of neem seed oil plus 10 milliliters of distilled water, we had a very low incidence of 28.6%. So which shows that the neem seed oil significantly at 4 ml concentration significantly reduced the incidence of the CMV virus. So the next slide is showing the severity of CMV. At week 1, it was not really, giving us it was really not stable. But when you check down from week 2, you will notice that at the control had a very high amount of severity, which is the light blue, what's it called? The light blue chart, bar chart. You see that it has a very high severity. Then coming down at 4 ml, we had a very low severity of 3.49. Moving to the 3 weeks after inoculation, the control is also showing a very high severity, and the next bar, which is the darker blue one, is showing a very low severity. Now moving down to the 4th week, you would see that the severity was about 8.44, which is very high. That is the control. That's to tell you that the neem oil, which was used as a control significantly reduced the severity of CMV. So the next slide is showing us the growth parameters. Plants established. Okay, plants that were established, we had 100% establishment, and numbers of branches. We took letters on numbers of branches, numbers of leaves, numbers of days to 50% flowering, numbers of leaves. And if you are looking at this table, you will notice that it significantly increased the numbers at 4 points. At 4 mil, there is a very high numbers in all these parameters as compared to the control, which is very small. That's to tell you that NIM is very effective in control of this virus, and it should be recommended. So at the next slide is showing the effects of different concentration of neem seed oil on the pepper yield. Now you would notice from this table that the yield was really increased. We had about 20.50 of amount of fruits. We have 20.50 numbers of fruits as compared to the control, which is about 10.50, which is almost half of that. So, and the weight of the fruits per gram is 148.0, which is relatively higher compared to the control and the other treatments that were used. So with this, I can say that neem seed oil has really helped during this experiment to control CMV virus, and I would say it should be recommended at the concentration of 4 mils. At the concentration of 4 mils, because a lot of researches have been carried out that shows the effectiveness of neem in controlling a lot of problems like they are antiviral, they are antifungal, they are antibacterial. But for this purpose of this presentation, I will be just focused on how it has helped to control CMV for this presentation. So thank you very much for listening to me, and thank you very much for the time to carry out this presentation.
Sure. Thank you very much, ma'am, for taking out your precious time to present your topic. Any questions, anyone? Yes, please. Please, sir. There's a question, ma'am.
Okay.
Yes.
Thank you, ma'am. Dr. Ali. Actually, you tell us, the audience, what are the causes of cucumber mosaic virus of pepper in Nigeria? And particularly, tell us about the climatic conditions of your country and date of sowing and harvest. And your slides are not visible actually. So that also could not grasp that thing. So on this point, if you will enlighten and highlight, then at least in Odisha will be much benefited. Thank you, ma'am.
All right. Thank you very much, sir, for that question. I don't think I got all the questions, but I think it's very hot over here. Our temperature ranges from 20 to 45. It's quite hot over here. And I think that was the only question I got, the one you asked on temperature. But the virus, I think I heard you said something about the CMV, the virus, the cucumber mosaic virus is a virus that started with cucumber, and we found out that it's not only affecting cucumber but also other vegetable crops like pepper, tomatoes, and which is of economic importance, that actually made us to start this study, this experiment. So I think I've been able to answer your question.
Thank you.
Hello?
Thank you very much, ma'am. Once again, let's have a round of applause for Dr. Ahmad Aliyu Santuraki. Thank you very much, madam, for joining us. Moving
Thank you, ma'am.
ahead, our next speaker is Priyanka Saha. Is she there with us? Priyanka Saha. She is not there. Dr. Sujoita Purohit. Dr. Sujoita Purohit from Mohanlal Sukhadia University.
Okay.
She is also not there. ବର୍ତ୍ତମାନ ମୁଁ ଚାଷୀ ଭାଇମାନେ ଯୋଉମାନେ ଅଛନ୍ତି ଭଉଣୀମାନେ ଅଛନ୍ତି ଆପଣମାନଙ୍କର ଯଦି କିଛି ପ୍ରଶ୍ନ ରହିଛି ଯେମିତିକି ଆପଣମାନେ ଜାଣିଛନ୍ତି 3 days କନଫରେନ୍ସ ଯାହାର ବିଷୟବସ୍ତୁ ଥିଲା କୃଷି ଖାଦ୍ୟ ସୁରକ୍ଷା ଏବଂ ପୋଷଣ ଦିଗରେ କିଭଳି ଭାବରେ କାର୍ଯ୍ୟ କରୁଛି ଖାଦ୍ୟ ସୁରକ୍ଷା ଏବଂ ପୋଷଣ ପାଇଁ କୃଷିର ଭୂମିକା ଏବଂ ଯେହେତୁ ଆପଣ ଗ୍ରାସରୁଟ୍ ମୂଳ ସ୍ତରରେ ମୂଳ ଭିତ୍ତିରେ କାମ କରୁଛନ୍ତି ତେଣୁ ଯଦି କିଛି ପ୍ରଶ୍ନ ଆପଣଙ୍କର ରହିଛି କିଛି ସମସ୍ୟା ରହିଛି ତାର ସମାଧାନ ଆପଣ ଜାଣିବାକୁ ଚାହୁଁଛନ୍ତି ଆମର ବିଶେଷଜ୍ଞମାନେ ଆମ ସହିତ ଅଛନ୍ତି ଆପଣମାନେ କିଛି ପ୍ରଶ୍ନ ପଚାରି ପାରିବେ ଷ୍ଟୁଡେଣ୍ଟସ୍ ମାନେ ମଧ୍ୟ ଅଛନ୍ତି ଯଦି କିଛି ପ୍ରଶ୍ନ ଆପଣଙ୍କର ରହିଛି ପଚାରି ପାରିବେ ଆପଣମାନେ जो विषय थिला विषय बाहर भी जदि आपणकर किछि भी प्रश्न रही किछि मनरे आपणकर समस्या रही जार समाधान सूत्र आपण जानवा को चाहूँगी आपण पचारि पारिबे हमर गवेषक माने अछंती विशेषज्ञ माने अछंती आ एई भली सम्मेलनीर मूल उद्देश्य आपण मानकर समस्यार समाधान आ खास करी आम चासी भाई भौणी मानकर आय को बहिर्भूत करबा पई द्विगुणित करबा पई आय वृद्धि करबा पई यह गोटे सुजोग आपणक पई यदि किसी प्रश्न रही आपण मैंने पचारि पारिबे वर्तमान मु अनुरोध करिबे हमर कन्वेनर सेशन कन्वेनर डॉक्टर एस के नाथ सेशन कोऑर्डिनेटर डॉक्टर ए के साहू आम सहित रैपिटेयर भाव में थे डॉक्टर एस पट्टनायक एंड डॉक्टर तपन कुमार पल्लई समस्त अनुरोध आम तांकठारू किछि जदि आपणकर समस्या रही तार समाधान विषय जानिबा आउ सेशंस गुडिक जे गुडिक रहिथिला तार रिमार्क्स मु रैपिटेयर को अनुरोध करिबी to please present and I would request the chairperson to kindly conclude with the concluding remarks. Please, sir. Please welcome our chairperson, Professor B.K. Sahu, advisor, SOA University Bhubaneswar, and Dr. Kishore Kumar Behera. Please, sir.
समस्त को मोर नमस्कार आमर सम्मानीय चाषी भाई माने माँ माने भौणी माने ये उपस्थित थिबा आमर मोर को चेयरमैन सेशन कोऑर्डिनेटर सबू रैपोर्टियर्स आउ आमर निमंत्रित अतिथि तापर आमर ଓଡ଼ିଶା କୃଷି ବିଶ୍ୱବିଦ୍ୟାଳୟ ସମସ୍ତ ଅଧ୍ୟାପକ ଅଧ୍ୟାପିକା କୃଷି ବିଜ୍ଞାନ କେନ୍ଦ୍ରର ସମସ୍ତ ବୈଜ୍ଞାନିକ ବୃନ୍ଦ ଏବଂ ଆମର ସବୁ ଛାତ୍ରଛାତ୍ରୀମାନେ। ତେଣୁ ଆଜି ଆମେ ସକାଳ 9:30ରୁ ସମସ୍ତେ ଏକାଠି ହେଲେ ଏବଂ ଆମମାନଙ୍କ ଗହଣରେ ସୁଦୂର ଆମେରିକା ନାଇଜେରିଆର ବିଭିନ୍ନ ବୈଜ୍ଞାନିକମାନେ ତାଙ୍କର ସବୁ ଭାଷଣ ରଖିଲେ। ଗୋଟେ ହେଉଛି ଆମେ କ'ଣ କଲେ ଆମର ତାଙ୍କ ନିଜ ଅନୁଭୂତି ସେ କହିଲେ, ତାଙ୍କ ଗବେଷଣା ଲବ୍ଧ ଜ୍ଞାନ ମଧ୍ୟ କହିଲେ। ତେଣୁ ମୁଁ ମୋର କୋ ଚେୟାରମ୍ୟାନ୍ ଡକ୍ଟର କିଶୋର କୁମାର ବେହେରାଙ୍କୁ ଅନୁରୋଧ କରୁଛି ସିଏ ଟିକେ ୟାକୁ ବ୍ରିଫିଂ କରିବେ, ତାପରେ ମଧ୍ୟ ମୁଁ ତାପରେ ମୋର ଏଇଟା କହିବି।
Jai Jagannath. Good afternoon, respected chair, session convener, my teachers in front of me, respected delegates, এবং আমার সমস্ত চাষী ও ভাই ও ভଉଣୀ ମାନେ, dear friends, and my colleagues present here. Thank you so much for participating in this deliberation, and also let me congratulate again and thanks all the presenters who have presented today. And let me take this opportunity to just share some of the highlights from the presenters, those who have already shared with us. And we had today 12 presenters, and out of that, I think 4 of them have not presented because of some challenges at their level, and rest 8 they could able to present their slides, and also we have put some questions for interactions, and they have also responded. Okay, sorry, so sorry, ମୁଁ ଓଡ଼ିଆରେ କହୁଛି, ମାନେ ଓଡ଼ିଆ ଇଂଲିଶ ରହିବ ମଝିରେ ମଝିରେ ୱାର୍ଡ କ'ଣ କରିବେ। ଆମର ଯୋଉ ପ୍ରେଜେଣ୍ଟର ମାନେ ଯାହା କହିଥିଲେ ସମସ୍ତେ, ତା ଭିତରୁ ମୁଁ ଭାବୁଛି ହାଇଲାଇଟ୍ସ କହିବି, ନା ମିସ୍ କରିପାରେ। ବଟ୍ ଆମର ଯୋଉ ଫାର୍ଷ୍ଟ ପ୍ରେଜେଣ୍ଟର ଥିଲେ ସେ ଆମକୁ କହିଥିଲେ କି ଲୋ ଟେମ୍ପରେଚର ପ୍ଲାଜମା କେମିତି ଆମର ସିଡ୍ ପ୍ରାଇମିଂରେ ୟୁଜ୍ ହେଇପାରିବ। ଆମେ ଯେତେବେଳେ ସିଡ୍, ମାନେ ସିଡ୍ ହେଉଛି ଗୋଟେ ଇମ୍ପୋର୍ଟାଣ୍ଟ ଜିନିଷ ଯୋଉଟାକି ଆମ ଏଗ୍ରିକଲଚର ବା ଆମ କ୍ରପିଂ ସିଷ୍ଟମ ଯଦି କହିବା ବା କ୍ରପ୍ ପ୍ରଡକ୍ସନ କହିବା, ତାହେଲେ ସିଏ ସେଥିରେ ଆମକୁ ବୁଝେଇଥିଲେ କି ଲୋ ଟେମ୍ପରେଚର ପ୍ଲାଜମା ଯୋଉଟା ଆଉ ଟିକେ ଭଲ। ଆଇ ଏମ୍ ଫ୍ରମ୍ ଏକ୍ସଟେନସନ ବ୍ୟାକଗ୍ରାଉଣ୍ଡ, କିନ୍ତୁ ଆପଣମାନେ ଆଉ ଟିକେ ଭଲ ବୁଝିଥିବେ ମୁଁ ଭାବୁଛି। କିନ୍ତୁ ସେ ଲୋ ଟେମ୍ପରେଚର ପ୍ଲାଜମା କେମିତି ଆମର ସିଡ୍ ପ୍ରାଇମିଂରେ ୟୁଜ୍ ହବ, ଆଣ୍ଡ ଏଗୁଡ଼ା କେମିତି ପ୍ରଡକ୍ସନ ବଢଉଛି, ପ୍ରଡକ୍ଟିଭ ବଢଉଛି, ଆମର ଫୁଡ ସେଫ୍ଟି ବଢଉଛି, ଆଣ୍ଡ ଫୁଡ ନ୍ୟୁଟ୍ରିସନ ବାବଦରେ ସେ ଭ୍ୟାଲୁ ଆଡ୍ କରୁଛି। ସିଏ ଆମକୁ ସେ ବିଷୟରେ ପୁରା ପ୍ରାଞ୍ଜଳ ଭାବରେ ବୁଝେଇଥିଲେ। କିନ୍ତୁ ଆମେ ବି ପଚାରିଥିଲେ କି ନାଇଁ ଏଗୁଡ଼ା ଫାର୍ମର୍ସ ଫିଲ୍ଡ ଗଲାଣି ନା ଏଗେନ୍ ଟ୍ରାଏଲ ବା ଏକ୍ସପେରିମେଣ୍ଟେସନ ବା ରିସର୍ଚ୍ଚରେ ଅଛି। ସେ କହିଥିଲେ ନାଇଁ ନାଇଁ ବର୍ତ୍ତମାନ ଆମେ ପରମ୍ୟୁଟେସନ କମ୍ବିନେସନ ଅଛୁ, ଆମେ ଟ୍ରାଏଲ କରୁଛୁ, ଏଗୁଡ଼ା ଫିଲ୍ଡକୁ ଆମର ଯାଇନି। ବଟ୍ ଇଟ୍ସ ବହୁତ ଭଲ ଟେକ୍ନୋଲୋଜି, ଯଦି ହେଇପାରିଲା ମତେ ଲାଗୁଛି ବହୁତ ଭଲ ହବା ଆଗକୁ। ବଟ୍ ଫ୍ୟୁଚର କ୍ୟାନ୍ ସେ ଓନ୍ଲି ଦିସ୍। ତାପରେ ନେକ୍ସଟ ଆମକୁ ନେକ୍ସଟ ପ୍ରେଜେଣ୍ଟର ଆମକୁ କହିଥିଲେ କି ଆମର ଯୋଉ ଦିଟା ୱାର୍ଡ ଡିକକ୍ସନ ଆଣ୍ଡ କନକକ୍ସନ, ଯେ କି ଆମେ କହୁଛେ ଯୋଉ ବେସିକାଲି ଏଫେକ୍ଟ ଅଫ ୟୁଜ ଅଫ ପେଷ୍ଟିସାଇଡ୍ସ, ଆମେ ଯୋଉ ପେଷ୍ଟିସାଇଡ୍ ବା ଇନସେକ୍ଟିସାଇଡ୍ ୟୁଜ୍ କରୁଛେ ବର୍ତ୍ତମାନ ତାର କେମିତି ହାଜାର୍ଡସ୍ ଏଫେକ୍ଟ ହଉଛି ସଏଲ ହେଲ୍ଥ ଉପରେ ବା ପ୍ଲାଣ୍ଟ ଉପରେ ବା ହ୍ୟୁମାନ ବିଙ୍ଗ ଉପରେ ଅଲ୍ଟିମେଟଲି, ଆଉ ଏଇଟା କେମିତି ଆଫେକ୍ଟ କରୁଛି ଏଣ୍ଟାୟାର ଜିନିଷ, ଯୋଉଟା କି ଶାଶ୍ୱତୀ ମ୍ୟାଡାମ ସେଟା ଭଲ ଭାବରେ ବୁଝେଇଥିଲେ କି ଆମେ କେମିତି ଅର୍ଗାନିକ ଆଣ୍ଡ ନ୍ୟାଚୁରାଲ ଫାର୍ମିଂ ୱେରେ ଆମେ ଚାଷ କରିପାରିବା, ସେ ବିଷୟରେ କହିଥିଲେ। ଜୀବାମୃତ ଏ ବିଷୟରେ ଆମକୁ କହିଥିଲେ କେମିତି ଗାଈର ଗୋବର ବା ଖତକୁ ଆମେ ୟୁଜ୍ କରିକି ଆମର ଫିଲ୍ଡରେ ଆମେ କମେଇ ପାରିବା ପେଷ୍ଟିସାଇଡ୍ର ୟୁଜ୍, ଆଉ ସେ ବିଷୟରେ ଆମକୁ ଭଲ ଭାବେ ବୁଝେଇଥିଲେ। ଆଣ୍ଡ ସେଥିରେ ବି ଆମେ ପ୍ରଶ୍ନ ପଚାରିଥିଲେ, ମ୍ୟାଡାମ ବି ବୁଝେଇଥିଲେ। ମୋର ମନେ ପଡୁଛି ରାଘବ ଭାଇ ମୋ ସାମ୍ନାରେ ବସିଛନ୍ତି, ଭାଇ ପଚାରୁଥିଲେ କି ନାଇଁ କେମିତି ହେଇପାରେ। ମୁଁ କହିଲି ଆପଣଙ୍କ ଡିପାର୍ଟମେଣ୍ଟର ପାଖ ଡିଷ୍ଟାନ୍ସରେ ଅଛନ୍ତି, ପରେ ବରଂ କଥା ହେଇପାରେ। ହୋପଫୁଲି ଦେ ୱିଲ୍ ଡୁ ଦ୍ୟାଟ୍। ଥ୍ୟାଙ୍କ ୟୁ। ଦେନ୍ ଆର୍ସେନିକ ଟଲରାଣ୍ଟ ଉପରେ ଆମକୁ ରାଇସ୍ ଫିଲ୍ଡରେ ରାଇସ୍ରେ କେମିତି ଆର୍ସେନିକ ଟଲରାଣ୍ଟ ବଢି ଯାଉଛି, ସେ ବିଷୟରେ ଆମକୁ ଡକ୍ଟର ତ୍ରିପାଠୀ ସାର୍ ଆମକୁ ବୁଝେଇଥିଲେ। ଆଣ୍ଡ ରାଇସ୍ରେ ଗୋଟା କେମିତି କମା ଯାଇପାରିବା, ଯୋଉ ଡୋନର ମାନେ ଗୋଟେ କଣ୍ଟ୍ରିବ୍ୟୁଟ କରୁଛି ତାକୁ କେମିତି ଆମେ ଏକ୍ସକ୍ଲୁଡ କରିପାରିବା, ସେ ବିଷୟରେ ସେ ବୁଝେଇଥିଲେ। ରାଇସ୍ର ଆର୍ସେନିକର କେମିତି ସେ କେତେ ଟଲରାଣ୍ଟ ବା କୋଉ ରାଇସ୍ ଟଲରାଣ୍ଟ, ସେ ବିଷୟରେ ଆମେ ବୁଝେଇଥିଲେ। ଆଉ କେମିତି ଆମେ ଆକୁ ଆଭଏଡ କରିପାରିବା, ବ୍ୟାଡ୍ ଏଫେକ୍ଟ ଅଛି ସେଇ ବିଷୟରେ ସେ ପ୍ରାଞ୍ଜଳ ଭାବେ ଆମକୁ ବୁଝେଇଥିଲେ। ଆଣ୍ଡ ସେ ପ୍ରେଜେଣ୍ଟେସନ ଆମେ ପ୍ରଶ୍ନ କରିଥିଲେ, ସାର୍ ବି ଡେଫିନେଟଲି ସେ ଟ୍ରାଏ କରିଥିଲେ ଆମକୁ ଅଡିଏନ୍ସକୁ ରେସ୍ପଣ୍ଡ କରିବା ପାଇଁ। ଆମର ନେକ୍ସଟ ଥିଲା କ୍ରପ୍ ହେଲ୍ଥ ମ୍ୟାନେଜମେଣ୍ଟ, ଯେ କି बायर बायोसाइंस रु जड़े तांकर साइंटिस्ट हमको क्रॉप हेल्थ मैनेजमेंट बेसिकली से प्रोडक्ट डेवलपमेंट बेले तांकर केमिति हमें जेनेटिक लेवल केमिति चेंजेस हमें कौन आनी पारे लुकिंग एट क्लाइमेटिक जलवायु जो परिवर्तन होउची जो क्रॉप को हमर अफेक्ट करूची ता विषय भी क्रॉप रे भी न्यूट्रिशन एंड आपरे अफेक्ट कर जो प्रोडक्शन अफेक्ट करूची प्रोडक्ट रे अफेक्ट करूची तो से कहथिले बायोसाइंस क्रॉप तांकर जो साइंस डिविजन अच्छी बेसिकली से फोकस करथिले राइस क्रॉप बा धान रे केमिति हमें परिवर्तन आनी पारिया प्रोडक्ट लेवल प्रोडक्ट डेवलपमेंट जेनेटिक लेवल जो गोटे कि हमें अमल रे किछि बाधा प्राप्त हबो नै बा न्यूट्रिशन लेवल किछि बाधा प्राप्त हबो नै फूड सिक्योरिटी सेफ्टी कथा भी सी हमको से प्रेजेंटेशन रे कहिथिले आ इनोवेशन कथा कहिथिले से माने केमिति करूछन देन अनदर आ गोटे इंटरेस्टिंग जो गोटे मैंग्रोव ऊपर कहिथिले जो गोटे कि हमर केंद्रापड़े जानथे मैंग्रोव रो बहुत माने बड़ बेल्ट अच्छी समुद्र कूल सल्ट टलरेंट नुहे किंतु सल्ट सार प्रेजेंटेशन दौले डॉक्टर सुमेन आई थिंक एंड सी आमको कहते कि सल्ट स्ट्रेस किंतु एक्चुअली अगेन सी भी कहते ना इट्स नॉट ए सल्ट स्ट्रेस कि सल्ट रे केमिति माने हमें सल्ट मैनेजमेंट ऊपर बेसिकली मैंग्रोव एरिया मैंग्रोव प्लांट के जो सल्ट एरिया सेटा फेवरेबल बा भलो हुए कि स्टिल देन सल्ट मैनेजमेंट ऊपर सी तांकर प्रेजेंटेशन रखिथिले जोटा कि वेरी इंटरेस्टिंग एरिया आमर भी कोस्टल जोन में किसी मैंग्रोव एरिया अच्छी आम किसी इनपुट नहीं आम काम करें कौट यूज हो पार ताल बहुत भल कथा देन आमको नाइजेरिया भी जेंडर लेंस रे आमको जड़े साइंटिस्ट बुझे सी कही कि केमिति हाउ टू इनक्रीज द पार्टिसिपेशन ऑफ वोमेन इन एग्रीकल्चर एंड जो भी आप देखिए नाइजेरिया कहीं आम स्टेट जब कह इंडिया कह ग्लोबली कह डेफिनेटली इट इज ए टॉपिक बर्निंग टॉपिक and बहुत दिन कहूँ किंतु केते एक्शन रे ट्रांसलेट होची दैट्स एवरीथिंग डिपेंड्स ऑन अस एंड द फार्मिंग कम्युनिटी किंतु डिसीजन मेकिंग हो जहाँ बा केमिति आमे सेमान पार्टिसिपेशन बढ़ाई पारे बेसिकली वोमेन मानकर महिला मानकर कि पार्टिसिपेशन बढ़ाई पारिबो सेउडा से कहथिले मोटामोटी आम देखिया इंटीग्रेशन ऑफ जेंडर बा जो महिला मानकर चाष रे जो सब इन्वॉल्व अछन कि तांको आमे केमिति प्रायोरिटी दिया हबो केमिति तांके से रिकॉग्निशन दिया हबो बा तांकर पार्टिसिपेशन बा भागीदारीटा केमिति बढ़ाई पारिबो से विषय रे बेसी हमकु वर्चुअली कहथिले नाइजेरिया रहिकरी तापर हमको नीम एक्सट्रैक्ट नीम सीड ऑयल एक्सट्रैक्ट विषय भी कहते केमिति हम कुकुम्बर सेटा यूज करि परिबा से विषय हमको जने साइंटिस्ट कहते हाउ टू मैनेज दिस डिजीजेस मत लगुच एतिकी प्रेजेंटेशन थिला समस्त ठारू जदिओ डिफिकल्ट थिला बेले बेले बाहरु हमको ठीक रे ऑडिबल हो नथिला स्टिल देन हम भाबुचे हमें ऑल द प्रेजेंटेशन दे हैव प्रेजेंटेड नाइसली एंड डेफिनेटली हम भाबुचे सब भी ज्ञान न पाइले भी एटलीस्ट we have been exposed to some of the technologies and researches or investigations or findings or innovations से दृष्टि देखिले हमरो भाबुचे किछि एथरा ज्ञान पाइचे एंड अगेन मु प्रेजेंटर मानु समस्त बहुत धन्यवाद दउची वी आर वेरी ग्रेटफुल टू द प्रेजेंटर्स थैंक यू फॉर द नाइस प्रेजेंटेशन एंड टेकिंग अस थ्रू ए जर्निंग ऑफ देयर इन्वेस्टिगेशंस और रिसर्चेस थैंक यू सो मच अगेन मु हमरो कोचर को धन्यवाद दउ सॉरी देन सुजीत को एंड एठी मोर टीचर माने बसते समस्त को मोर प्रणाम एवं समस्त को अगेन मु धन्यवाद देकरी मोर रहउछ थैंक यू सो मच
आमर ये जो सप्तम अंतर्जातीय जो आमर जो सेमिनार तार यहाँ शेष टेक्निकल सेशन थिला या परे आमर उद्यापन उत्सव हो एवं आज जो आपण मैंने एत संख्या समत एकाठी होती मुझे शुणिली आम सुंदरगढ़ सुदूर सुंदरगढ़ चाषी भाई मैंने माँ भौणी मैंने समस्त आसुँच आम समस्त को कहवाई सुदूर अमेरिका नाइजेरिया तो विभिन्न वैज्ञानिक माने बहुत भल भल जिनस कहले जोटा कि आम ताकू शुणा ग्रहण करवा एवं खाली सेतिक नुहे आम जो केवल आम रासायनिक कीटनाशक हो कि आम रोगपी हो आम जो सब औषध व्यवहार करचे ताकू किछि परिमाण ह्रास करी आम मैंने विभिन्न प्रकार आम जो देशीय जिनस अच्छी ताकू व्यवहार करी केमिति आमे भल परिवेश को संतुलन रखा से विषय शुणिले एवं सभा शेष आमर निम्ब तेल आम केमिति ताकू निम्ब तेल प्रयोग करी आम किपरि रोग को प्रतिरोध करा से विषय शुणिले खाली सेतिकी नुहे आमे जो कहूचे नारी शक्ति से नारी शक्ति पे मध्य आम जड़े वैज्ञानिक से बहुत गवेषणा करते तेणु यहाँ आम गोटे भल जिनस खाली ओडिशा नुहे भारत वर्ष नुहे सुदूर नाइजेरिया से करते नारी शक्ति को आम देखुचे बहुत माँ भौणी अच्छा तेणु आम ग्रहण करवा तो मुझे प्रथम धन्यवाद देवी प्रथम आमर रापोर्टर जो मैंने अच्छा मोर अनुरोध से जो जिनस गुड़िक से रेकर्ड कर गोटे प्रोसेसिंग आकार दबा एवं मोर को चेयरमैन डॉक्टर बेहरा सेशन कोऑर्डिनेटर हमको धन्यवाद देवी सेशन टी बहुत भल भाव यहाँ को परिचालन होगा तापर आम जे मैडम आमको कहते प्रत्येक कथा अनाउंस करते मो तरफ बहुत धन्यवाद धन्यवाद।
ग्रुप फोटोग्राफ नबा किंतु ता पूर्वरु किछि क्वेश्चंस जोटा पछरे डिस्कस करबा बोली हमें कहिथिले एबे अरुंधति मैडम अछंती मैडम को मु अनुरोध करबे मैडम आसीबे एवं जहां प्रश्न थिला तार उत्तर रखिबे प्लीज मैम। जोरदार करताली सहित स्वागत जणेबा डॉक्टर अरुंधति सासमल। सर आपनकर प्रश्न रहिथिला चाहिबे आपण पचारिबे मैडम को आज्ञा। आज्ञा चासी भाई भौणी माने जो माने अछंती आपनकर जहां भी प्रश्न रहिछि आपनकर समस्या संबंधी प्रश्न मैडम को आपण पचारि परिबे कृषि विशेषज्ञ अछंती आम सहित मु चाहिबे आपनकर प्रश्न आपण माने रखिबे के किछि प्रश्न पचारिबा पई चाहुचंती मु जानिबा पई चाहिबे आम सहित केते जण चासी भाई अछंती टिके हाथ ऊपर को करिवे। जदि आपण किछि प्रश्न चाहुचंती पचारिबा पई मैडम को पचारि परिबे। मैडम रहिबे प्लीज मैडम गोटे ग्रुप फोटोग्राफ पई आपण समस्त को रिक्वेस्ट करिवे आसीबे ग्रुप फोटोग्राफ मैडम प्लीज जॉइन फॉर द ग्रुप फोटोग्राफ। आज्ञा आज्ञा आज्ञा। अच्छा प्रथम आमर जेतिकी चासी भौणी माने अछंती मु अनुरोध करिबे आपण मानकु आगु को आसीबे हम विशेषज्ञ माने अछंती हमर स्पीकर्स माने अछंती तांका सहित आमे गोटे ग्रुप फोटोग्राफ नबा। प्रथम महिला चासी माने जेतिकी जण अछंती प्लीज आगु को आसीबे आसंतु आसंतु। अच्छा प्रथमे सर मु भाबुची हमर महिला चासी जेतिकी जण अछंती प्रथमे तांको सहित गोटे ग्रुप फोटोग्राफ जेतिकी जण महिला माने एठी अछंती चासी भौणी माने समस्ते आगु को आसीबे गोटे ग्रुप फोटोग्राफ। केते जण चासी भौणी एठी अछंती टिके हाथ ऊपर को करिवे प्लीज। आगु को आसीबे प्लीज समस्ते आपण माने दया करी आगु को आसीबे गोटे ग्रुप फोटोग्राफ। सर आगे लेडीज मानक संगे गोटे फोटोटा दरकार पड़िबो आगु। प्रथमे सबु महिला चासी माने दया करी प्लीज आगु को आसंतु आपण माने एवरीवन पछरे जो माने अछंती ऑल द लेडीज हां अधा जण प्लीज तळे छिडा होई जिबे। सर आगे ये फोटोटी गोटे नहीं जिबा convener for the session, coordinator and the rapporteurs, please come and join. एबे ये फोटोटी नहीं जिबा भाई प्लीज आसीबे सर या पारे पुनी थरे स्टूडेंट्स आसीबे जॉइन करिवे समस्ते आसुचंती फ्रेम रे आपण दुई जण शेष रे प्लीज टिके सेपटको पळेबे समस्ते फ्रेम रे आसुचंती हां एबे लेफ्ट साइड रे जो माने अछंती समस्त फ्रेम रे आसीबा दरकार तो आधा आधा जण आसीबे प्लीज लेफ्ट साइड रो आधा जण आसंतु प्लीज समस्त फ्रेम रे आसीबे आधा आधा जण करिके आसंतु सर मैडम सर प्लीज ऑन जो माने ऊपर अछंती प्लीज रहिबे ऑल द प्रोफेसर्स प्लीज जो माने बसी चंदी लेफ्ट साइड रो आधा जण प्लीज आगो को आसीबे फोटो फ्रेम रे समस्त आसीबा दरकार आधा आधा जण करिके आसीबे समस्त Professors are requested to kindly step back. Sir, please रहिबे मैडम स्टेज ऊपर जो माने अछंती प्लीज रहिबे आपण माने टिके आगो को आसीबे प्लीज गोटे ग्रुप फोटोग्राफ अच्छा जो माने बसी चंदी समस्त एबे टिके आधा आधा करिके आसीबा आगो को स्टूडेंट्स ऑल द स्टूडेंट्स ଆଉ ଯେତିକି ଚାଷୀ ଆସି ନଥିଲେ ଯେତିକି ଚାଷୀ ଆସି ନଥିଲେ ଆଉ ଷ୍ଟୁଡେଣ୍ଟସ୍ ମାନେ ପ୍ଲିଜ ଆସିଯାନ୍ତୁ, ଅଧା ଅଧା କରିକି ଆସିବେ, ଅଧା ଅଧା ଜଣ ଆସିବେ, ଫଟୋ ଫ୍ରେମ ରେ ସମସ୍ତେ ଆସିବା ଦରକାର, ଏକା ବେଳେ ହେଇପାରିବନି। ଟିକେ କ୍ଲୋଜ ପଳେଇ ଆସିବେ। ପ୍ରେମ ରେ ଏତେ ଜଣ ଆସିପାରିବେନି, ଟିକେ କ୍ଲୋଜ ପଳେଇ ଆସିବେ, ଅଧା ଅଧା କରିକି ନେଇଯାଆନ୍ତୁ ଭାଇ, ଏପଟୁ ଅଧା ସେପଟୁ ଅଧା। ଆଉ ଯୋଉ ଚାଷୀ ଭାଇ ମାନେ ରହି ଯାଇଛନ୍ତି ପ୍ଲିଜ ଟିକେ ଆଗକୁ ପଳେଇ ଆସିବେ ଫଟୋଗ୍ରାଫ ପାଇଁ। ଥ୍ୟାଙ୍କ ୟୁ ସୋ ମଚ୍, ଥ୍ୟାଙ୍କ ୟୁ ଅଲ୍ ଦ ପ୍ରଫେସର୍ସ। या परे उद्यापनी समारोह अनुष्ठित हब, मु आपनମାନଙ୍କୁ अनुरोध करिबि आपण माने समस्ते हॉल रे रहिबे। उद्यापनी समारोह परे लंच एरिया ओपन हब, वैलिडिक्टरी परे लंच एरिया ओपन हब। आमर गेस्ट माने उद्यापनी समारोह रे आम सहित सामिल हेबे आ किछि समय रे ही तुरंत आमे आरंभ करिबा उद्यापनी समारोह वैलिडिक्टरी सेरेमनी। आई वुड रिक्वेस्ट ऑल ऑफ यू टू काइंडली take your seats. Please be patient and please wait. Thank you. Folder टा कहूँ सर पठेवा पे। Thank you.
ତିନିଦିନିଆ ହ୍ୟାଲୋ 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 ତିନିଦିନିଆ ଆନ୍ତର୍ଜାତିକ ସମ୍ମିଳନୀ ର ଶେଷ ଦିବସ ଏବଂ ଶେଷ ସମୟ ସମସ୍ତଙ୍କୁ ଆମେ ସ୍ୱାଗତ କରୁଛୁ। ବହୁତ କମ ସମୟରେ ଆମର ମୁଖ୍ୟ ଅତିଥି ପହଞ୍ଚିବେ, ତାପରେ ଆମେ ଆରମ୍ଭ କରିବା ଔପଚାରିକ ଭାବେ କାର୍ଯ୍ୟକ୍ରମ। ହଁ ଶେଷ ସମୟ ସରିବ।